Well, good morning, everybody. Those of you that are here, the few of you that are here, and the many more that are online. So welcome to you all. About 2 years ago, we decided we would have this meeting. I'll explain about that in a minute. Thought at the time it would be a much more local affair. We might get 40 or 50 people. And in fact, by the time we decided we couldn't have it, this was Easter last year, we had about 30 odd registrations, I think. We thought we might get 40 or 50. Things moved on. We moved it a year. We still couldn't have it. And eventually it landed up now. And fortunately, and really thanks to the university, I'm very grateful to people in the university who have made this happen for us. We're able to have this event both in person, which is really nice. I really like actually being able to see people for real as well as on screen. And it means that now we have, I don't know what the final figure will be, but well over 100 people, probably 120 people, either logged in or here, and people from all over the world. So welcome to all of you, wherever you are at the moment. I hope you're going to find this meeting very interesting. I think we've got a really interesting program there, and it will tell you something about what's going on in disease and pest management within the UK. So why is the AFCP involved? But the Agri-Food Charities Partnership is set up to try and make sure that agricultural charities within the UK talk to each other and can understand what each other's doing, work together wherever possible, and therefore benefit UK agriculture and beyond. So that is what we're there for. This particular meeting is because, in fact, a number of charities, and you'll see their logos coming up quite frequently on the slides in future, have been funding work on rape, diseases and pests. And actually quite a lot of it at this university. So this is a great place to host it. And you'll hear much more about that later. So thank you to all those charities that have been funding work, and I hope will continue to fund work here and elsewhere on disease and pests in rape.
Yeah, good morning, and apologies for the poor communications during that the session this morning. I'd just like to introduce the Agricultural Universities Council in this very brief presentation. I'm Rob Edwards. I'm the head of the Natural Environmental Sciences School at Newcastle University, which encompasses all of the agricultural sciences. And I'm representing the Agricultural Universities Council here as their acting chair. So first of all, what is the Agricultural Universities Council? Well, it was an organisation that we conceived in 2018 when the Agriculture Bill was published. And although the Agriculture Bill talked about skills, it made very little mention of the role of the universities, though it did make specific mention of the further education colleges. And suddenly, together with other things that were ongoing in the scene, in the UK farming scene in that time, we recognised the fact that actually the agricultural universities do not have a concerted voice, and they don't have a place really at the table for any of the decisions that are being made about skills development, either in government and also to an extent within industry. So we thought we would get together as a group of universities to try and change that persona of us being a group of fractured academic institutions and speak instead as one organisation. So in terms of the membership, we have all of the UK universities represented with agricultural schools and departments, which are actively involved in education and research in agricultural related programmes, and particularly those which are linked to food and environmental land management. And you can see there's a fairly extensive list here. We haven't included universities which just have veterinary schools, but all of the universities here are members, whether they're post-92 or pre-92. And if we've left anyone out, that's only because of oversight rather than any particular motivation. In terms of the vision of what we're trying to do here, we see the UK really needing to have a leading agri-food sector driven by high quality scientific research and education, and where better to get that from other than the university sector. And as a mission, we would like to develop a central role for the universities in the global future of the agri-food sector in the UK. So our our ambitions are not just national, indeed they're driving the UK agri-food sector internationally. The aim is to represent HE University interest in agriculture and related topics and speak with one collaborative voice to industry, government and other stakeholders. We often hear, particularly from government, that the many disparate voices of universities that they encounter are not helpful in terms of representing the value of university research and training to government. So they would much rather that we acted as one. And this is a step on that particular path. The primary objectives of the AUC are to provide a forum for agri-food thought leadership. So, we come from the university sector, we should be able to innovate and come up with new ideas and new approaches. We also want to pool resources to enable evidence-based information and commentary context the agri-food related policy, preferably not contradicting ourselves endlessly. To work together to ensure a pipeline of high quality graduate researchers and leaders in the UK and international agri-food sector. So that's something everyone recognises the value of, but actually tangibly, there isn't a single organisation that represents that skills base and how we develop it at a national level. We're also interested in working with other organisations to complement and add value to existing initiatives and groups, and to facilitate and raise the profile of agriculture as a very important science-led subject at a national level. Activities in 2020 were pretty much brought to a close in March. We did have a meeting early in 2020 and we're pretty much ready to go to the next stage. And because many of us are involved in running large schools, large management units, it was felt appropriate that we really could pause the activity of the Agricultural Universities Council until the pandemic was under control. We have undertaken some of the work that we started on. We started an audit of current education and research facilities across the member institutions. And in 2019, David Llewellyn and others were actively working with the Food and Drink Sector Council, planning on agri-schools development and the role of universities within that. We need, as our next step, to consolidate our organisation into the establishment of a secretariat with a website and press communications. We're trying to do that as an independent organisation, supported by an independent organisation, rather than have it affiliated with one university that has a number of legal issues which actually tied us up for some time trying to resolve. We have engaged with industry and government. We've met with the DEFRA Chief Scientific Advisor. We also work very actively with the Food and Farming Futures Group and Lord Curry and colleagues. So we can see, of course, a lot of our interests overlap with Lord Curry's, and clearly we want to be able to work with them rather than in competition with them. So next steps, we will resume our activities later on this year. We'll form up our secretariat and recommence engagement activities. Many of our institutions are hosting chairs or other positions which are funded through the Elizabeth Creek Charitable Trust. And through that trust, we have formed a research network, and those chair positions in particular, funded through the Elizabeth Creek Charitable Trust, we see as a very important resource in terms of providing leadership to this particular Agricultural University Council group. Later on this year, we intend to host a workshop from the AUC on university-led research and innovation and its impact on the agri-food sector. So really raising the importance and profile of university research in driving forward many of the innovations that we're currently enjoying in developing in agriculture and indeed in the wider food chain. We are, of course, open to suggestions to how we can work with other organisations such as other charitable trusts. And that was one of the reasons I think that Bruce Fitt and others thought it would be a really good idea for us to come along and talk to you today. So I'm sorry about the technical difficulties we encountered, but I hope you've got something out of this presentation which will show you effectively where we're going with it all. And at that point, I think I shall stop the presentation. And thank you very much for your attention.
My career food path can be divided into 3 stages. The first stage is from 1991 to 1999, when I was at the Oil Crops Research Institute in Wuhan, China. The second stage is from 1999 to 2011, when I was at Rothamsted Research. The third stage is when I was at University of Hertfordshire since 2011. At the first stage of my career, I mainly worked on stem rot, because stem rot is a major disease problem on oilseed rape in China. My work at that time mostly focused on crop physiology, cultural practice, chemical control, and breeding in related to stem rot control. At the second and the third stage of my career, I mainly worked on former stem canker and the lettuce spot. Former stem canker, also called black leg. So former stem canker and lettuce spot are two major diseases of oilseed rape in the UK. The symptoms of former stem canker and the lettuce spot can be observed on leaves. So the top is former leaf lesions on the leaf, and second is the lettuce spots. And also the symptoms can also observed on stems, and the stem canker and the lettuce spot on stem. And also the symptoms can be observed on pods as well. These two diseases caused ear losses more than 100 million pounds in the UK. So for susceptible cultivar, plants can co-infected by the two diseases. So this, you can see the picture of the plants infected by former leaf spot and lettuce spot. The co-infection by these two diseases caused the big ear losses. For former stem canker, I will talk about three areas. First, I talked about pathogen biology and epidemiology. And second, I talk about the world distribution and UK-China collaboration. And third, I talk about host resistance. In the UK, former stem canker caused by two closely related pathogens, Aermacillans and Aerbacillus. To make it easier to speak, I call AM and AirB. In autumn and winter, AM cause big former leaf spots with the pycnidia, the bottom one, you see with the dark dots. And the Bacillus cause small dark lesions with no or few pycnidia. In summer before harvest, Bacillus mostly associated with upper stem lesions, and AM mostly associated with stem-based canker. Therefore, AM is considered more damaging than AirB. These two species coexist on their host. They have a similar life cycle. The epidemics of former stem canker started in autumn from ascospores, released from pseudocysia, developed on the infected debris. You can see in the pseudocysia, the mature pseudocysia release ascospores. You can see in the hole on the top. After spores released, the ascospore landed on leaf surface, caused former leaf spots. Then the fungi can grow along the leaf vein and leaf petiole to the stem. In summer, they cause stem canker. This is the cause of lodging, and they cause the main cause of ear losses. You can see for this life cycle, let's only have one cycle within the crop season. So it's also called a monocyclic disease. So from this life cycle, you can say control this disease is mainly need to focus on these four stages. Ascospores of AM and AirB, the size and shape are similar. It can't be distinguished. However, after germination, they can be distinguished. Ascospores of AM, the germ tubes normally started from the middle of the ascospores, and the hyphae grow tortuously with the branches. And ascospores of AirB, the germ tubes normally started from the two ends of ascospores, and the hyphae grew straightly and with few branches. These two species also can be distinguished when grown on the PDA. AirB produce a fluffy colony and with yellow pigment, and AM, they grew very flat colony without pigment. However, the morphology and the pigment production is not always reliable to distinguish these two species. So the distinguish confirm these two species, I have to use species-specific PCR. Epidemics started by ascospores. To investigate how long ascospores can survive after release, we investigate at 4 temperatures. Surprisingly, ascospores can survive quite a long time. For example, at 20 degrees, after release, 35 days is about 10% of ascables still alive can germinate, as about 2% of AFB ascables can germinate. As in the four tested temperatures, you can see the AM ascables survived longer than AFB ascables. To investigate whether there are differences between AM and AFB in ascable maturation and pseudocystia development, so we also tested 4 different temperatures. At 15 and 20 degrees, you can see the pseudocystia maturation rate for AM and AFB are similar. There's no significant differences. However, at 5 degrees and 10 degrees, AM ascables matured faster than AFB ascables. Because the former stem canker epidemic started from ascables, no ascables, that means no former leaf spores. There's no need to spray fungicide. Therefore, it's important to monitor ascable release and build a model to forecast ascable release for guiding fungicide application. For example, this photo you see, this picture, this is the pattern of ascable release, and then the black dot, the round dots is the incidence of former leaf spore. And then the triangle is the incidence of former stem canker. You can see in the early October and August, you see there's little or no ascable release. There's no former leaf spores. When the ascable started release in the late October, November, lots of spores release, you can see the increased incidence of former leaf spore. Therefore, the timing of ascable release is very important for fungicide spray because the early release of ascables leading to early infection and leading to severe canker if you not controlled. So first of all, for former stem canker, I mainly talk about the distribution in the world. In Australia, former stem canker mainly caused by AM. But in Canada, USA, Europe, is former stem canker caused by both AM and AFB. However, in China, it's only AFB detected so far. There is a need to prevent AM from spreading into China because Chinese cultivars are very susceptible to AM. You can see this picture. This is our field trial last year. A Chinese cultivar is very susceptible, severely infected by former stem canker and caused lodging, where the British UK cultivar stand still is very strong, still very good. And also, pep choy is very popular brassica vegetable in China. You say when they grow in the UK, you say get full of the former leaf spore. So that's no use. So you can imagine if AM spread into China, they will cause big economic losses to Chinese oyster rape and Chinese brassica vegetables. To prevent AM from spreading into China and be prepared for control former stem canker epidemics in China, we developed a collaboration with China with the 5 institutions. For example, the Shenyi Liu's group in Oil Cross Research Institute, Professor Guo Qingli and Meng Jinlin group in Huazhong Agricultural University, and Professor Ziqing Li's group in Inner Mongolia, and Professor Bao Chunhu's group in Anhui Academy of Agricultural Sciences, and also Professor Xianping Wu's group in the Institute of Plant Quarantine of China. So the collaboration monitored funded projects by Royal Society Fellowship or Royal Society International Joint Project, and also China Scholarship Council funded scholarships, and also British Council funded projects, and therefore UK funded the same project. For effective control of former stem canker by host resistance, we relied on 2 types of resistance. First is major gene-mediated qualitative resistance. The second is minor gene-mediated quantitative resistance. So you say former stem canker started from a leaf infection. So if there's no leaf infection, that means there's no growth of fungi from the leaf to the stem. To choose this, we use R gene-mediated resistance. But if we still have a leaf infection. That's a second mechanism. So if we get an infection on the leaf, so we need a mechanism to prevent or slow down the growth of the pathogen along the leaf to the stem. And if once the pathogen reaches the stem, and also can prevent or slow down the growth in the stem and to reduce stem canker severity. You can say at the top you get a severe stem canker and the bottom one got a less severe canker. You got a less severe canker, you get a less impact on the yield. To achieve this, we use quantitative resistance. So this is the picture taken from field. In the field in the UK, drug without no effective R genes, you get 4 infection by the pathogen, you get 4 loss of formative spores, and the cultivar DK extrovert have effective RM7. You can say the growth of the pathogen is restricted at the infection area by the resistant gene. So the pathogen can't grow from leaf to the stem. To investigate how R gene prevent the pathogen growth from leaf to the stem cause stem canker, what we did, we inoculated the cultivar with RM6 with the isolate labeled with GLP to see how the pathogen grew in after infection of the leaf. You can say after inoculation, the growth of the pathogen, you can see the green fluorescence, the GLP is the hyphae of the fungi, they are restricted at the inoculation site. Therefore, they can't grow from the leaf to the stem. After the inoculation leaf dropped off, you can see from the leaf scar, there's no canker developed. On the right-hand side, you can see this confirmed by GLP, by the efflorescence, there's no green fluorescence there. However, for the cultivar without effective R genes, inoculated with the GLP labeled isolates, you can see the fungi can grow along the leaf, from the inoculation site along the main vein down the petiole to the stem. And after the inoculation leaf dropped off, the canker developed at the leaf scar and also confirmed by the GLP. So R-gene-mediated resistance is complete resistance. It's easy to assess at the seasoning stage. You can say either the resistant or susceptible. But R-gene-mediated resistance is risk-specific. For example, for the cultivars with carrying R-gene RM1, inoculated with the macrolens carrying the corresponding around near of the effector gene AV9 to RM1, you got a resistant phenotype. Otherwise, you get a susceptible phenotype. In the natural conditions, after the large area use of R gene, they give the selection pressure to the pathogen. They can mutate from around to variant. If they mutate from variant to around to variant, then the RM1 is not effective. Therefore, the R gene-mediated resistance is very easily to render ineffective. So they need to monitor the pathogen population for effective use of R gene resistance. So this example of a breakdown of RM6 mediated resistance, this is a field experiment done in France. You can see in the first and the second year, the RM6 mediated resistance is very effective. They get less severe canker. However, in the third and the fourth season, the resistance breaks down because the pathogen population changed from avirulent to virulent. A last example is for monitor the pathogen population for effective use of RM1. This is work done in Australia. So in 2001 to 2003, the frequency of avirulent of around RM1 was predominant. At this time, if you use the cultivar carried R gene RM1, it's very effective. However, after the large area use of the RM1, cultivars with RM1, the pathogen population was changed from avirulent to virulent, and the frequency, you can see 2006 and 7, 5 and 6, you can see the frequency of avirulent of avirulent RM1 is reduced to less than 40%. At this time, if you use cultivars RM1, it's not effective. So it's very important to monitor pathogen population for effective use of R gene resistance. To determine if avirulent genes in the isolates, we use the coordinating inoculation. We inoculate the isolate to a differential set of cultivars with a lone R gene. For example, if you locally isolate onto the Bristol carried RGM2, if you got to the resistant phenotype at the top, then you say this isolate carried a variant near of the corresponding variant RM2. Otherwise, they carried the virulent. In this way, we can characterize all the isolates as different effector genes. So this is the changes in frequency of aviaries in the RM population in the UK over 4 years. You can say the frequency of a variant near of 1, 2, 3, 4, and 9 are very low. This means the corresponding R gene arm 1, 2, 3, 4, and 9 are not effective in the UK. The predominant variants in the UK population is mainly 5, 6, and 7. However, there's no commercial cultivars with arm 5 and 6 are available. In the UK, most cultivars we use the effective R gene is arm 7. However, you see the frequency of a variant nears of arm 7 is decreased within the 4 years. So that means there's a risk of breakdown of this resistant gene. Similar situation was observed in France. You can say the frequency of variant alleles against the arm7 is increased over the 10 years in France. In some area of France, cultivars with arm7 is not effective anymore. So now I move on to the quantitative resistance. Quantitative resistance at leaf stage, whether you have quantitative resistance or not, you got a formal leaf support. Therefore, quantitative resistance also called partial resistance. At the end of the plant stage, if you cultivar with quantitative resistance, it gives less severe canker. If without quantitative resistance, you get severe canker. So quantitative resistance means can reduce the severity of stem canker and is before the harvest and is not a phase-specific. Quantitative resistance is considered more durable. So to understand how quantitative resistance to reduce prevent the growth or slow the growth of pathogen to reduce the severity of stem canker. So again, we inoculate the leaves with the GOP labeled isolate. So let the fungi grow to the stem, cause stem canker. We use the two cultivars, Damor with quantitative resistance, Ura without quantitative resistance. So when you inoculate the leaf, the fungi can grow to the stem, can cause canker on both Damor and Ura. However, after the cross-section, you can say the stem canker symptoms mainly the cortex of the stem of the Damor, but spread into the piece of Ura. This is also confirmed using GOP observation. So once the fungi spread into the piece and the stem piece, and then they weaken the stem because the woody bit is damaged and cause lodging. So quantitative resistance is mainly slow down the growth of pathogen from the leaf to the stem and also slow down the canker development and prevent the growth of the pathogen from the outside the cortex to the piece and eventually reduce the severity of canker before harvest. So current effective use of R gene control from stem canker in the UK is mainly use RM7. However, isolate virulent against the RM7 already detected, so I need to develop strategies to prevent or avoid breakdown of this important resistance. Also, there's a need to investigate new sources of resistance. Because former stem canker caused by two related species, the RM and RB, control of former stem canker either by fungicide or by breeding need to target both RM and RB. So now I move on to lettuce spot. Lettuce spot recently become a major disease problem in the UK. You can say this is the yield losses by 4 diseases in the UK. The red bar is yield loss caused by lettuce spot. You can say the yield losses by lettuce spot increased recently. Unlike former stem canker, lettuce spot is a polycyclic disease. Epidemics also started in autumn by asc spores released from epicenter developed on infected debris. The asc spores landed on the leaf and germinated and can penetrate directly through the cuticle, and they grew for long-term symptoms list. And the symptoms can be observed in spring. You can see with sporulation. You can see in the white dots, aquae with loss of canidia. By rain splash, they can spread into other parts of the plants and also close by plants, cause secondary infection. Furthermore, within the season, infected synthetic infected leaves can develop epicenter, and the epicenter released from this developed on the dead leaves cause the second infection in spring. So within one season, they have several cycles of the pathogen, making it very difficult to control. Lettuce spores have a long period of symptomless time. So infection normally in the field observed in the autumn, but the symptoms are not available until spring. Sometimes the severe symptoms are not available only after incubation. For example, this leaf one in the field, you don't think it's very severe. However, after incubation, you can say the whole leaf is covered by the pathogen, so the whole leaf is infected. Host resistance against lettuce spot pathogen is less understood compared to former stem canker. Currently, research mainly focuses on cultivar E-mono. The resistant phenotype is observed also like black flecking along the main vein or leaf petiole, also on the leaf, without pycnidia, without sporulation. And susceptible cultivar, you say, with lots of sporulation. So the resistance in the E-mono observed is mainly stopped. We think mainly stopped a sexual spores sporulation, and because no asexual spores, and then they can reduce or stop the secondary infection. The major gene loci derived from the cultivar E-mono mapped on the chromosome A1. However, there is more work to do to investigate these resistant genes and develop markers for breeding, and also to understand the mechanism of resistance. Furthermore, there's very little work or information about quantitative resistance against lettuce spore pathogen. So, control lettuce spore pathogen is very difficult and very challenging because lettuce spore is a polycyclic disease, and they have a long symptomless period after initial infection. And the host resistance is less understood, and there is little knowledge about the variation of the pathogen populations. Furthermore, the development of fungicide insensitivity was already observed in this pathogen. So, host resistance is ever more important for control this disease. I would like to thank the founders and the collaborators for funding different projects during my career in the UK, especially the different industry partners and also agricultural charities. I also would like to thank the colleagues, especially Bruce, for his support and encouragement during my career in the UK. I also would like to thank other colleagues at the University of Hertfordshire and my previous colleagues at Rotham Research and international collaborators in China, France, Poland, Australia, Canada, and also previous and current PhD students at Rotham Research and at the University of Hertfordshire for their help, support, and friendship. My special thanks go to 4 members of AFCP for their funding for my PhD and also for funding of my 8 PhD students. I co-supervised Georgia, Thomas, and Katie. I am principal supervisor for Asna, Harika, James, Laura, and Evan. When I was preparing my presentation, I realized in a few days, it will be 21 years since I gave my first presentation in English to Perry Foundation trustees. I convinced them to support my PhD project. This is a group photo after my presentation. You can see everybody is very happy. I have a very happy memory. Thank you very much for your attention. Okay. Okay. Okay.
Maybe others here, but we've got time for one other one, I should think.
Okay.
Right. So the question was, overall, how many R genes have been identified for stem canker? I mean, is it just the ones you've listed, or are there others?
Oh, stem cancer morning. I think morning 12 genes, and two of them cloned.
And are they specific to one species of Leptosphaeria, or?
Yeah, they're specific to the or species specific R genes. One is called Leparm3 and RM2, but actually they are near or the same gene.
Okay. Are there any other questions from the floor? If not, then thank you very much for a very interesting talk, and obviously a huge amount of work there over many years.
Thank you.
So our next speaker, James Fortune, has got an interesting title. It's called Chemical Warfare, the Fungal Quest to Conquer Oilseed Rape. Thank you, James.
Hello everybody, my name is James Fortune and my presentation is called Chemical Warfare, the Fungal Quest to Conquer Oilseed Rape. So first, foamy stem canker is caused by two similar coexisting pathogens, Leptosphaeria maculans and Leptosphaeria biglobosa. And as you can see on this image here, their lesions look very different depending on whether they're on the leaf or on the stem. So on the leaf, L. biglobosa forms a smaller, more defined lesion, whereas on the leaf, L. maculans forms a much larger, paler lesion. And then if we look on the stem, L. biglobosa seems to be formed further up the stem, and they're called upper stem lesions, whereas L. maculans is formed further down on the stem, in the stem basal region. The difference in their stem location has been suggested to be due to differences in their ascospore maturation and therefore their release dates. But L. maculans and L. biglobosa can be found in the stem basal lesions, and I'll come on to a potential reason for this later. So just a few foamy leaf spots. Well, since 2009-2010 growing season, foamy stem canker has caused annual yield losses worth more than £74 million on average to the English and Welsh growers. As you can see here, it's quite an economically damaging disease in terms of millions of pounds. But this is all despite growers using resistant cultivars and effective fungicides. Shifting of spores. So this is what I was coming on to, what I explained earlier. So studies have shown that L. biglobosa, previous studies have shown that L. biglobosa ascospores were released later than L. maculans. However, more recent investigations that have used qPCR analysis have reported that ascospores of both species are more frequently released at similar times. Here's an example of this from my field trial up in Kings Lynn. So this was Terrington St Clement up in Kings Lynn, Norfolk in the UK. So as you can see here, the red line represents L. maculans, but the blue line represents L. biglobosa. And if we look at this image or this graph, L. biglobosa L. biglobosa ascospores were released at the same time as L. maculans, which is very different to what previous studies have shown. But also, it's released in a higher concentration. So this is something that may explain, in terms of that release date, why L. biglobosa and L. maculans are found at the stem basal lesion as well. So the interesting title, the chemical warfare. So L. maculans does produce a non-host selective epipyrithiodioxypiperazine, or an ETP for short, and that's called cyrodesmin or cyrodesmin PL. Here's the image of its compound structure, and it's characterized by the disulfide bridge in the top right-hand corner, the disulfide. L. biglobosa does not produce this compound, but cyrodesmin has been shown to have an inhibitory effect on L. biglobosa. And cyrodesmin has been shown, literature has shown that cyrodesmin takes between 3 to 5 days to be produced at a detectable level. And this was what I've shown in similar results have been shown by myself in the lab. So the aims of this study were to investigate the interactions between L. maculans and L. biglobosa, as well as the changes in phytotoxin production as a result of increased interspecific competition. So how did I do this? So we had liquid culture where I put fungal plugs of the two diseases, L. maculans or L. biglobosa, either in isolation, so L. maculans only, or L. biglobosa in isolation, so LB only. But also I put them both in at the same time, and that is L. maculans and L. biglobosa simultaneously inoculated. They were then placed in a rotary shaker, and then after 2 weeks, the culture, the mycelial mass was filtered away from the culture filtrate, and then the secondary metabolite from the culture filtrate were extracted using ethyl acetate, and then that was done through, and then the assessments were done either through a bioassay or a HPLC analysis with further mass spec. So the results. So the effect of secondary metabolites on colony growth. So as you can see here, we've got 2 rows and 3 columns. The top row represents L. maculans, the bottom row represents L. biglobosa. And the first column is ethyl acetates, that's the control. So that is what the secondary metabolites were extracted in. And we've also got L. maculans only, so they're the extracts from the L. maculans only, and L. biglobosa only, and they're the extracts from the L. biglobosa only. So as we can see on the L. maculans, in terms of the colony growth after 7 days was not affected by any of these different different extracts, sorry. Whereas L. biglobosa, there was a significant inhibition of growth when the L. maculans only extract was applied. So that's what it says there. So the secondary metabolites produced by L. maculans inhibit the L. biglobosa colony growth, and that supports previous literature. But when we put the isolates from, the extracts from L. maculans and L. biglobosa, this inhibition was lost. This was rather surprising, but this is something that now I've got the extracts and the HPLC analysis, I can identify what the differences were. So this is the results section for the detection using the HPLC analysis. So the pink line represents, this is off a HPLC chromatograph. The pink line represents the peaks that are found in the L. maculans only. The blue line represents what's in the L. biglobosa only, and the green line for L. maculans and L. biglobosa at the same time. But what is really interesting, there were 3 unique peaks that were only found in the L. maculans only. So that was peak 1, or maximum 1, maximum 2, and maximum 3. So these are where the pink line, where there's a peak that's on the pink line, but not on the other 2 extracts that we assessed. So yeah, there were 3 unique maxima. So the identification of the precursors. So a sample was taken from maximum 1. So here's the mass spec output from here, and we found there was a peak at 445.2. So that represents a compound with a molecular weight of 444.2. And there's a known compound called diacetylcyrodesmin PL, which has a molecular weight of 44.5. So we're quite confident with that. And also there was an additional peak at 381, which is actually could relate to diacetylcyrodesmin lacking the 2 sulfur atoms. So that's a common thing within this term of mass spec. But also we found an additional peak at 319, which relates to a compound of a molecular weight of 318 daltons. And that relates to another compound that's known as known secondary metabolite of L. maculans, and that's fomaamide, which has a molecular weight of 318. So we're quite confident that that is also being represented in this spectra. And both, as I've said previously, both of these were known secondary metabolites, but interestingly, both are precursors for the formation of the compound cyrodesmin that I explained earlier. So when we looked at the maximum 2, we found a peak at 487.6, and cyrodesmin, funny enough, has a peak of 486.6 as well. We also found different adducts, so we've got the 423.27, which could correspond to the cyrodesmin lacking the 2 sulfur atoms, as well as additional adducts at 504, and which relates to the ammonium adduct of cyrodesmin, as well as 509, which relates to another sodium adduct, sorry. So we took this one step further and we started to look at the interaction in plantar. So if you look in the top left-hand corner, we've got the sterilised distilled water. Sterilised distilled water. So all we did was we grew cotyledons and inoculated using a pin, inoculated with sterilised distilled water, or L. macculans only, Conidia, this is all Conidia, L. globosa only, and then the combination of the two at the same time. So what we can see here is L. maculans forms a very large lesion, a large pale lesion, which is very similar to what is seen in the field, as well as L. globosa, and then L. globosa forms a much smaller, more concise lesion, which is similar to what's also seen in the field. But interestingly, when L. maculans and L. globosa were inoculated at the same time, they form very similar lesions to what was seen, or in phenotype, very phenotypically similar lesions to L. globosa, suggesting that L. maculans has been outcompeted by L. globosa. So, competition between, and then we took this one step further. So we took leaf sections from these lesions and then extracted the DNA to examine the difference in the DNA concentrations between the different treatments. So the amount of L. maculans DNA and L. globosa DNA were reduced when co-inoculated. So if you look at this graph here, L. maculans, as you'd expect, there's a lot of L. maculans DNA. L. globosa on its own, you'd expect lots of L. globosa DNA. Whereas when we combine the two, there was a reduction in both, but the reduction was disproportionate. There was a much, L. maculans DNA was much more reduced than L. globosa, as we can see here. So 95% reduction compared to the L. maculans only, and 40% reduction compared to the L. globosa only. There's been many suggestions that there's the LB can prime L. maculan, prime the plant to L. maculans, but that would sort of infer a defense response. But why is this so disproportionate? There must be something else going on, and that's where I think this interaction between the two pathogens is coming into play. So this is work that needs to be done further. So in summary, the secondary metabolites, cyarodesmin and its precursors, are produced by L. maculans, inhibit the colony growth of L. globosa. There were 3 unique maxima, so cyarodesmin precursors and cyarodesmin PL in itself, and that third peak we were unable to identify, so that's remaining unknown. L. maculans and L. globosa are co-inoculated, L. maculans and L. globosa are co-inoculated cyarodesmin. The precursors are not produced. So when they're co-inoculated, these precursors are not produced, and therefore the actual product of cyarodesmin cannot be produced. So how does this sort of relate to the real world? Well, guidance in from the AHDB and other sources suggests that fungicides, typically azole fungicides, are applied when there's 10 to 20% of the crop incidence of L. maculans like leaf spots. So if L. maculans and L. globosa astrospores are released at the same time, foamy leaf spot lesions may appear smaller or later, resulting in a later application of fungicide that would, a later application of fungicide which would affect the control of this disease and other diseases. So I'd just like to thank everybody I'd like to thank everyone. So Dr. Yongzhu Huang, Professor Bruce Fitt, Dr. Faye Ritchie, Dr. Chintony, Karen Denny Douage, as well as the picture hasn't turned up, but also Dan Baker and James Stanley. I'd also like to thank all of my funders for all their help and support with this project. So the Chad Acre Agricultural Trust, Hertfordshire Local Enterprise Partnership, University of Hertfordshire, Felix Thornley Cobbold Trust, ADAS and the European Union. And thank you. Is there any questions?
So there's one question. So there's one question come up on the screen from Kerry Maguire. I think actually it came up just at the end of Yongdu's talk, but you could probably answer it as well. If not, she can come up and have a go. Looking at LM and LB, will one become dominant, do you think, as varieties change? And as perhaps with climate change, are we likely to see any change in populations?
Well, that's a very interesting question. Thank you for that question. It's very much an unknown. It's very much unknown, but increase in temperature does. Sorry, when I was going back to the astrospore release about the potential reasons for why L. maculans and L. globosa are released at similar times, the pseudothecial maturation is affected by the environmental temperatures, and L. big globosa at temperatures above 10 degrees are mature faster, so therefore the release might occur earlier, and that's why we're getting these results. So coming back to your question about that, we don't know, but L. big globosa is definitely on the rise, and we are seeing this more frequently, and certain papers from France have also seen this. I don't know if Yongju's got anything you'd like to add on this matter.
If you have, you better come up here and say it, Yongju, because oh, there we are. We've got a microphone.
Yeah, I think the changes of LB populations recently, we observed the increase of LB definitely, and also the seems related to the weather conditions as well. It's very interesting. Some year we got very high proportion for LB identified, and other years very less. So we are still doing some research to investigate whether the weather conditions. Also, you see, I presented about the temperature effects on the maturation of the two species. So this is the one we are going to investigate, and also fungicide application as well, and also the use of the R gene, especially ARM7. The use of ARM7 controlled AM, leading to the increase of the LB. So there's definitely different factors.
Thank you. Okay. Are there any other questions from the floor? Ah, there's one up there. Got a mic coming.
Hi, just wondering if you could theorize as to why when you grow LB with the extract of LM, why it inhibits the growth, but then when you inoculate on the plant, then it seems that LB inhibits LM.
Right, so that comes down to, if I just quickly go back to the slide, or I won't actually. So it comes down to the inhibition of that syrodesmin. So in the plant, so that's in the plant, they're being extracted at the same time. In the plant, they're being inoculated at the same time, and that's why I mentioned about the time of delay between the production of syrodesmin. So that if they're at the same time, it's before the syrodesmin can be produced. So therefore, L. big globosa can get the advantage. Whereas when we're putting the extracts onto the L. big globosa fungal plug, the syrodesmin is already being produced. So therefore, it's having the inhibitory growth, the effect there.
Okay. That's it. Good. Well, thank you very much.
Thank you.
So we, having thought about canker there, we're now going to think a bit more about light leaf spot, disease management in oilseed rape, insights into the Brassica napus perina brassicae pathosystem. And our speaker is Chintani Kandeni Duwaja.
So, hi everyone, my name is Chintani. I am a research fellow here at the University of Hertfordshire. So my main research interests are understanding host pathogen interactions in major oilseed rape pathogens, mainly for my stem canker and light leaf spot. So today I'm going to be talking to you about light leaf spot to provide some insights into the interactions between Pinna brassicae and Brassica napus. So light leaf spot is currently the most damaging disease of oilseed rape in the UK. With annual yield losses of more than 120 million pounds. The pathogen affect the crop at various growth stages, infecting leaves, stems, flowers, and pods, causing substantial yield losses. Also, the pathogen affect a variety of vegetable brassica species, again causing yield losses and also affecting the quality of this vegetable produce. Just really a little bit about the pathogen. P. brassicae is a ascomycete fungal pathogen. The disease starts in autumn with airborne ascospores that land on leaves of the young oilseed rape plants, and the pathogen colonizes in subcuticulus spursus. So we can see the first symptoms of the disease in late winter or in the spring as lesions associated with white crystal-like structures. So these are the asexual sporulating structures of the pathogen we call acervally, and they can further infect the crop through rain splashes, cause secondary infections, and this is also one of the main causes for the occurrence of lightly spot epidemics. So after harvest, the pathogens survive in crop debris, undergo sexual reproduction, and produce inoculum for the next cropping season. Current management of oilseed rape relies on application of fungicide and host resistance. The timing of fungicide applications has always been a problem with lightly spot because of the long asymptomatic growth phase of the pathogen. So there are some disease forecasting systems to assist farmers with fungicide application timing. Also, there are resistant ratings available for lightly spot, oilseed rape cultivars for different regions in the UK. These are available through AHDB recommended list trials. So other than that, there has been some cultural control practices suggested or recommended, including plowing of oilseed rape debris, crop rotations, or maintaining a distance between previous and current oilseed rape fields. However, looking at the development of the epidemics over the past years for lightly spot, we can clearly see that these available control strategies are not always effective, or they are not adequate to contain the disease. So we need better control strategies. For example, the fungicide applications may not always be an economic solution for the farmers, especially small and medium scale farmers. And also there is the risk of development of fungicide insensitivity due to the reproductive potential of this pathogen. Also, there is very limited understanding of the genetic basis of resistance against this pathogen, especially if we compare that to the knowledge we have about the other major pathogens like former stem canker. We know very little about P. brassicae, even though it's present since 1970s with frequent epidemics. So other than that, as I mentioned, the P. brassicae has quite large host range. So there are evidence of cross-infectivity of this pathogen between oilseed rape and vegetable brassicas. Also, cruciferous weed species can act as a host to this pathogen. So these other species can actually provide a sort of pathway for the pathogen to move from one crop to the next cropping season, despite whether there are cultural control strategies or not. Also, there have been some records on breakdown of host resistance. So what you can see here is more recent record of resistant breakdown in cultivar cracker. This is a recommended cultivar for the North region in the UK with high resistant rating. And what you can see here in the picture is heavily stunted plot of cultivar cracker because of the breakdown of resistance. And you can, if you can compare that to the normal height crop next to it, you can see the effect of this pathogen on these cultivars. And this graph shows some early examples of resistant breakdown for two cultivars, cultivar Apex and Bristol. In these graphs, the gray bars represent the market share of these two cultivars. As you can see here, both the cultivars seem to have a good level of resistance in the beginning. However, when the widespread cultivation of these two cultivars occur, the status of the cultivars has shifted from resistance to become susceptible. So that means we need very robust sources of resistance to contain this disease. So our research here at the University of Hertfordshire has mainly focused on resistant and susceptible phenotypes identification, differential interactions, and understanding genetic and molecular basis of resistance against P. brassicae, and also identification of new sources of resistance so that we can provide better cultivars with good resistance against lightly spot to better management of this disease. So looking at various symptoms associated with disease, there has been previous records on the about P. brassicae affecting the growth regulators of the plants, causing various deformations in the plant. So we observed the similar scenario in our control environment experiments. The interesting fact here is that these are the first visible sign of the disease appear as early as 7 days after inoculation under controlled conditions. And also, interestingly, we could see these symptoms on both resistant and susceptible cultivars. Again, P. brassicae does not normally produce immediate hypersensitive response similar to other extracellular fungal pathogens. However, there are records of necrosis appearing in cultivar Imela that considered to have a major gene for resistance against P. brassicae. And in our control environment experiment, we also observed this type of necrosis appearing around 9 days after inoculation in various other host genotypes. Sometimes these necrotic flecking appear together with some amount of sporulation of the pathogen. So there seems to be some continuative nature into this. So this is an illustration to show how the disease development might occur between a resistant and a susceptible host plant. All the resistant sources that we know to date does not actually prevent initial infection or the colonization of the pathogen. So we think the appearance of leaf deformations in both resistant and susceptible plants could be associated with this early infection phase or the early colonization phase of the pathogen. And that could be the reason why we see it in both resistant and susceptible host plants. However, as the disease progresses, we can see the host recognition of the pathogen, and it tries to, or it limits the further colonization of the pathogen, and also ultimately it prevents the asexual sporulation of the pathogen. So if you can see here in the top right-hand picture, you can see there is a black flecking in a resistant response, and in the bottom right-hand picture, you can see the sporulation or the acervally in a susceptible interaction. We also looked at the possibility of differential interactions between P. brassicae and Brassica napus. So for this experiment, we use 18 different oilseed rape cultivars and lines, consist of commercial cultivars, breeding lines, and also we included parental lines of some mapping populations. And of course, we include cultivar Imela as a reference point for resistance. And we inoculated this, all these genotypes in a series of glasshouse experiments using single spore isolates of P. brassicae, and then monitor the disease development and did the disease scoring or the severity assessment using percentage leaf area covered with P. brassicae sporulation. So this looks a little bit complicated graph, but I will take you through different steps. So basically this shows how each of these different genotypes respond to different isolates of P. brassicae. As I mentioned, we use cultivar Imela as a point of reference for resistance, and then we try to identify other resistant interactions. So those other resistant interactions are the ones that you can see as marked with asterisks. And you can see there are some cultivars that has isolate-specific resistant interactions. Also, we saw that some of the breeding lines have quite consistent resistant response throughout almost all the isolates. So these could actually be some good sources to be used in oilseed rape breeding programs. Talking about parental lines of the mapping populations, these two cultivars Dharma and Udal, which are the parental lines of DY population, produce the most contrasting difference between the resistant and susceptibility. So this is something that we need to further look into and could also provide some other potential sources of resistance. Now moving on to the opposite side of the scale, moving towards the susceptibility. This is very interesting. These three cultivars, Excel, Harper, and Hearty, rank the highest in terms of their susceptibility. The interesting thing is all these three cultivars are the very resistant cultivars against the other oilseed rape disease, former stem canker, and all three of them are known to have RLM7 resistant genes against Leptosphaeria maculans, which is highly effective in the UK. So this is something we really need to look further into And if you look at this all very familiar graph now, this poses the need for some cultivars that are resistant against several pathogens. Because as you can see here in the graph, the light leaf spot yield losses caused by light leaf spot has increased over the years. But former stem canker has also been a persistent disease problem throughout these years. So we really need cultivars that can perform equally against both of these pathogens. And this is another example taken from one of our field experiments. And again, as you can see here, cultivars that are better performing against stem canker are susceptible to light leaf spot and vice versa. So what we actually need is cultivars like DK cabinet that perform equally against both the diseases. Having said that, we also looked at some other sources of resistance. Brassica napus is known for its narrow genetic diversity because of the strong selections that occurred in its breeding history. But being a natural hybrid between Brassica rapa and Brassica oleracea, as you can see in the bottom two pictures, it allows us to make synthetic crosses between these two species to bring in new features into cultivated Brassica napus. So we looked at this Q double haploid population, which is a synthetic cross between Brassica oleracea and Brassica rapa. This is fairly a small population consisting of 92 DH lines. So what we did is first we phenotyped this population using glasshouse experiments. We used 3 different glasshouse experiments and also used different P. brassicae populations originated either in England or in Scotland. So this confirmed us that there is a resistance for light leaf spot segregating in this population. And you can see with reference to other resistant cultivars like Imela or susceptible cultivars like Marathon or Charger. Some of these lines seems to be performing better. So these are some of the phenotypes that we observed in those glasshouse experiments. So again, we saw these necrosis in some of these lines. And if you can see the pictures, 2 pictures in the bottom left-hand side, you can see there's a susceptible cultivar, and you can see the acervoli as salt crystalline. The picture next to it shows black spots. So this also again confirmed that this resistance somehow prevent the pathogen from producing the acervoli. However, there are other lines that had limited sporulation without any necrosis. So that was also interesting, and it suggested there could be 2 types of resistance segregating in this population. So because of that, we looked at the lines with or without presence of necrotic flecking. And from these 2 graphs taken from 2 experiments show that the lines that produce necrosis had significantly less sporulation than those that didn't have any necrosis. But there's also a variation in the amount of sporulation, even if there is no necrosis in those lines. So this is quite interesting, and there could be some quantitative resistance involved in it. And based on that, we could separate these lines into 4 different categories. There are a small number of lines that produce no sporulation, no necrotic flecking. And there are also a small number of lines that produce no sporulation with flecking, but majority of the lines had some amount of sporulation either with or without the presence of necrosis. Again, that could be due to the presence of any quantitative trait loci. So we also did some QTL mapping using this data. Here I'm showing some of the preliminary results of this QTL mapping. The interesting thing is there seems to be several QTL specifically in the C genome or mostly in the C subgenome. This is also interesting because if you consider the resistant sources that are integrated from ancestral Brassica species, most of them seem to have originated from the A genome, but in this case we can see most of the QTL in the C subgenome of Brassica napus. So That brings to the summary of my presentation. So we can see there appear to be some specific resistant interactions between P. brassicae and Brassica napus. And also there seems to be two main types or phenotypes of resistance. Those are the necrosis or the limitation of pathogen sporulation. And also there is a need to produce cultivars that perform equally against both former stem canker and light leaf spot to sustain oilseed reproduction in the UK. And also the secondary gene pool of Brassica napus may provide new sources of host resistance. However, we still need to identify quite a lot about the pathogen population variation, especially in Scotland and in England, to see if there are any variation of these pathogens, and that could help us how to deploy cultivar resistance effectively if there are any population structures or structural variations of pathogen populations. And also we need to look further into cross-infectivity between different Brassica species. So I would like to thank Professor Bruce Fitt, Dr. Henrik Storz, Dr. Youngju Huang, and all the other members of the Crop and Environment Protection Research Group at the University of Hertfordshire, and also Coretta Kloppel for all those nice pictures of vegetable brassicas that you saw in this talk, and all the funders and collaborators in my research. And yeah, thank you. If you have any questions, please do ask.
So thank you for that. One question came in, which I think you've sort of partially answered really anyway, and it was really to do with the resistance and varieties breaking down, and it's linked with novel races and how much we understand about those new races.
Yeah, I think that's the area we really need to look further into. If there are resistant breakdowns, that means definitely there could be some structural variations in these pathogen populations. So if you are to recommend any cultivars for different regions, we need to have a clear understanding of what sort of population structures are there in those different regions. So there is like very little information on those regions, and we really need to look further into that to see, you know, whether there is or how big is the population variation between different regions.
Okay, thank you. Is there any questions from the floor? No, in that case, thank you very much.
Thank you.
So now we change, and we're changing from pathogens to pests, and the pest that I think has been on every oilseed rape grower's lips for the last few years, certainly in the UK, really because of the ban on some pesticides. And this is obviously stem flea beetle. The talk is called Potential of Biopesticides and Optimizing Use of Conventional Insecticides for Control of Cabbage Stem Flea Beetle. And I'm going to ask Claire Huaro to come and talk about it. This is, while she's coming on stage, this is from a charity point of view, it's an interesting project. We have 5 charities actually involved in funding this project, along with the AHDB. And this is one of a number of projects which we're working on that way at the moment. So over to you, Claire.
Hi, everyone. Thank you, Graham, for introducing me. So I'm Claire Huaro, and I'm a second-year PhD student at Harper Adams University. And so yeah, I'm focusing on biopesticides to control the cabbage stem flea beetle in our seed rape crops. My project is funded by CERTIS, AHDB, and the AFCP, which I think. And I'm happy to present my work at the AFCP Forum, Management of Oilseed Rape Disease in Pests today. So the project aim and context. So the need is to identify alternatives to conventional insecticides, because as we know, the European Union has banned the use of nicotinoid insecticides on flowering crops in 2013, so it includes oilseed rape. And it means that the control of the cabbage stem flea beetle has been relying on pyrethroid insecticides only. However, most of the cabbage stem flea beetle population in the UK are not resistant to pyrethroids. And so pyrethroids are basically not efficient anymore. And pyrethroids are also harmful to non-target organisms such as parasitoids and predators of cabbage stem flea beetle. So there is an urgent need for alternatives. And the one I'm focusing on is biopesticides such as entomopathogenic fungi, bacteria, nematodes, and also plant extracts and physically acting products. So I've done several bioassays with entomopathogenic organisms, and the first one I'm going to present is the entomopathogenic bacteria Bacillus thuringiensis subspecies Tenebrionis, which has shown to be efficient against other species of Coleoptera. So I've tested different formulations that are still in development at field rate. So CEU 4077 IVG and CEU 4078 IVG from Certis, and INBS 32 from Endomat. So the bacterial solution was prepared at field rate, and then I dipped oilseed rape leaves, which were used as a food source for cabbage stem flea beetle adults. I replicated each treatment 6 times, and I also added a water control to compare. And I've recorded the cumulative mortality of the adult beetles every day until 12 days post-inoculation, as Bacillus thuringiensis can be a slow-acting pathogen. Just a quick word about how does Bacillus thuringiensis works. So it infects the host insect by ingestion. So it's a bacteria that sporulates and produce a crystal that contains delta endotoxins. And once the bacteria has been ingested, this crystal will dissolve and release the toxin, which will then bind to receptors in the midgut of the insect. And this binding will lead to the creation of pores in the midguts, in the cells, and the host will basically die of septicemia within a few days of ingestion. This is so the cumulative mortality of cabbage stem flea beetle after 12 days post-inoculation, and we can see that I didn't get any mortality above 40% of dead cabbage stem flea beetle after 12 days. So after these results, I've decided not to investigate entomopathogenic bacteria any further. Next, I've done a bioassay with entomopathogenic nematodes, 4 species in particular. Steinonema felthiae, Steinonema capocapsae, Steinonema crossae, and Heterorhabditis bacteriophora, which are nematode species that have quite a wide host range, but are known to infect other Coleoptera species such as weevils, garden chafer, and other species of flea beetles. So I've used commercial formulations from BASF of nematodes that come into as a paste to be dissolved in water, and then I would take 1 milliliter of the stock solution and then count the nematodes in this solution to determine then how much to dilute the solution to obtain 3 different concentrations. So 4,000, 10,000, or 40,000 nematodes per milliliter on water. Replicated 3 times for each treatment, and of course a water control as comparison. And I've recorded cumulative mortality every 2 days until 8 days post-inoculation. Entomopathogenic nematodes work as infective juveniles, who are the infective stage of the nematodes, the 3rd juvenile stage. So they are the only stage that is that can live outside of a host, and that's actively search for a host. Once they have located it, they will enter by a natural opening or by piercing the cuticles. Then once in the hemocell, they will release a bacteria that is contained in their gut and with which they have a symbiotic relationship. So the bacteria will then reproduce in the insect and will kill the insect and dissolve the inside of this body. And then all these material will provide food for the nematodes, which will then grow into adults, then reproduce. And then the infective juvenile will again get out of the host and search for a new one. So these are the results. So we can see here that the species Heterorhabditis bacteriophora is the one that worked the best, with more than 75% of mortality after 6 days at the middle dose. Then Steinema felsiae worked also quite well at the highest dose, with after 6 days more than 100% basically mortality. Steinema capocapsae worked quite well as well, especially at the middle dose and the highest dose, but it was slower to kill, only reaching high numbers, high percentage of mortality after 4 days. And the species that worked less well was Steinema crossae, that didn't reach more than 75% of mortality, but all of this is still better than the control, so quite encouraging results. And then I've also did bioassays with physically acting product. So with FLIPA, which is developed by Bayer. It is a fatty acid-based product that is currently authorized in horticultural protected crops against whiteflies, aphids, and mites. And another formulation by Certis in development, CEU 4064 OISL. I've tested at 3 different concentrations, half the fill rate, and 2 times the fill rate. For the methods, I've prepared the solution and then I've sprayed directly onto the beetles and the oilseed rape leaf as food and support. As it is a contact insecticide, the beetle has to come into direct contact with the product. It was replicated 3 times again, and I recorded cumulative mortality after 1, 2, and 5 days, as it's a quick acting product. This is an extract from a Bayer website about FLIPA to explain a bit what it is. So it is a product made of a carboxylic acid, which have a lipophilic carbon chains that can bind to the insect cuticle and then penetrate the insect cuticle. And it will then disturb vital processes and kill the insect within typically 24 hours. Those are my results. So we can see the control was great, no death at all. And it's quite nice result with FLIPA, with 100% mortality at double the field dose, even after just 1 day, and more than 85% mortality at the field dose after just 1 day again. So I'm quite happy with those results. And CEU 4064 ISL performed a bit less well, but still much better than the control. So quite encouraging results. I was happy with those results with FLIPA, so I did them again a few weeks ago. And this time I kept cadavers of cabbage stem flea beetles, and I tried to inspect the cuticle through a scanning electron microscope to see if we could visibly see the effect on the cuticle. And we can see on the right that the unity of the scales in the cuticle seems to have been disrupted compared to the control on the left. What next? I still have a few biopesticides that I want to test, especially entomopathogenic fungi. So I have some commercial formulations of Metharrhizium bruneum and Bovea bassiana lined up. I'm also going to test new formulations of physically acting products in the lab, and I'm also going to compare those biopesticides with conventional pyrethroid insecticides as a positive control. As with as what I did with Flipper, I'm going to see if other biopesticides have an impact on cabbage stem flea beetle cuticle through the scanning electron microscope. I'm also planning to investigate the persistence of all those biopesticides on oilseed rape leaf. So for the liquid product, to determine how long the leaves will stay wet, and for the microorganisms such as nematodes and fungi, how long does the spores and nematodes stay alive on the leaf, and what would be the density on the leaf. And all I've done so far is mostly lab work, and in September, to start my third year, I'm going to do field trials with selected biopesticides in the lab to see so that it reflects better real cropping formulations, cropping situations, sorry. And later in my PhD, I will also explore the cost and benefits aspects of the selected biopesticides to see if it would be feasible for farmers to actually use them in the field. And I will also test the selected biopesticides on non-target organisms to make sure that it won't be dangerous to use them in the field. So thank you very much for your attention. If you want to find out more about my project, you can follow me on Twitter. And there is also on AHDB website and CERTIS website some information about my project. Thank you.
So thank you, Claire. Haven't got any questions come up on the screen. Does anybody in the audience like to ask a question? Yeah, I've got one here.
Yeah, I imagine in a field setting the nematode treatment would be a little bit more tricky than the Flipper treatment. So how many times do you think the treatment would be repeated, and how long the nematode could survive on the leaf surface, let's say?
Yeah, all of these products are going to be applied several times. I tried to do a single plan for all of them, but I came up with 4 applications every 7 days. So even the physically acting products are recommended to be applied several times. So it would not be only for nematodes. For the survival on the leaf, that's something that I'm planning to determine before the field trials. And as they are living organisms, they are sensitive to UV lights and also to humidity. So the plan would be to apply them in the evening or very early in the morning so that the humidity is optimal. And also to keep an eye on the weather forecast, so that it doesn't get washed off the leaves. So yeah, we're going to plan to find the better conditions for that. Yeah.
Nematode, is that specific then for the cabbage stem flea beetles, or affects other coleopterans as well?
Yeah, it affects they affect many species, but they do affect some coleopteran beetles and also other species of flea beetles that are quite similar to cabbage stem flea beetle. So in the lab, it works quite well, and I'm hoping that in the field it will work well as well.
Okay.
Thank you.
Thank you. Any other questions? Okay, thank you, Claire.
Thank you.
We've got a few minutes, and I saw that a couple more questions come in on light leaf spot, which I think are for Yongju, really. They're genomic ones. Yeah, if you'd like to. So the first one, is the genome being sequenced, and if so, when's it going to be available?
Which genome sequence?
Sorry, this is the P. brassicae one.
Yeah, we sequenced, but I think it maybe take about at least 6 months. We are preparing the paper.
Right.
Yeah, maybe 6 months time.
Okay.
In 6 months time, yeah.
Right. You're going to commit yourself to that, are you?
Hopefully, yeah, because things move very slowly, especially during the COVID situation.
Okay. And the second one is to do with QTLs, and are the QTLs for canker and light leaf spot at the same loci, and if so, are they difficult to combine if you're trying to get resistance to both species?
We haven't compared the QTL from the resistant against the Formosan canker and the light spot yet. But we used, also just mentioned, we used the same mapping population with the DY population in collaboration with France. We identified QTLs, use that population for Formosan canker. And that population also used in the one year in field experiment in France. They identified some QTLs. But Qintani also reported she's using the Q population as well. So we want to try to compare different QTLs, use different population to try to identify the common QTL. So hopefully it's useful for breeding. And we're not sure whether they overlap, the same QTL overlapped for resistant against Formosan canker pathogen and also against the light spot pathogen. If we can find that kind of QTL, that will be very useful for breeding.
Okay, thank you. Isabel Sims is going to talk to us and about resistance to soil-borne Rhizoctonia in oilseed rape. Thank you, Isabel.
Hello. My name's Isabel and I'm currently doing my PhD at the University of Nottingham, and I'm doing a doctoral training programme there, so I'm just in my third of 4 years, and I'm actually currently working for the HDB doing my 3-month placement. But today I'm going to talk about my PhD. And my purpose is to see if we can identify some resistance to Rhizoctonia solani in oilseed rape. My primary supervisor is Rimiana Ray. So what is Rhizoctonia solani? It's a soil-borne necrotrophic plant pathogen, and it has a really wide host range. So as well as affecting things like brassicas, it also affects cereals, potatoes, coffee, a huge wide range of species. And this can be really problematic for rotations. It also is really difficult to control. Currently, we use a range of cultural and chemical methods. So the weather is known to play a big impact, as well as things like tillage and things like that. Seed treatments are really commonly used, but as we know, a lot of chemicals get banned, so we need to make sure we have other methods as well. And there's currently no known resistance to Rhizoctonia in oilseed rape. You can see the 2 pictures I've got there. One is under a microscope, so it has really characteristic right angle branching with a slight constriction at those branch points. And then Rhizoctonia, it grows in the soil and it produces sclerotia, which you can see on the plate, which are really small sort of brown masses, and they will remain in the soil for a really long time, further sort of giving more problems for management. It splits into anastomoses groups, which affect different crops in different ways. AG21 is particularly pathogenic to oilseed rape, and that's what I'm interested in. So the hypotheses I'm going to discuss this afternoon are that although there's currently no known resistance to oilseed rape, in oilseed rape to Rhizoctonia, I hypothesize that there will be some differences in the phenotypes that we see. And based on other research to necrotrophic pathogens, I'm anticipating that it's more likely to be linked to jasmonic acid and ethylene responses, and salicylic acid might be involved in susceptibility. We also know that other AG groups of Rhizoctonia have been shown to produce PAA, which is a type of auxin. So auxin might also be linked to susceptibility. So the first thing I did was look at some different varieties under inoculation. I grew them for 7 days, and I grew them in hydraulica, which is like small clay pellets, so that then I could take out the roots really clearly. Because it's a soil-borne disease, a lot of the symptoms are on the roots. So if they're just in compost, it can be really difficult. You often destroy all the roots when you pull it out, and then you can't really see what's going on. Here is a nice little symptom scale. So 0 is completely healthy, and 4 is completely dead. You can see there's like necrotic lesions, damping off. The plants really suffer quite a lot. A lot of plants do die within 7 days under inoculation. So these are the results for the symptoms of 5 varieties that I tested. And you can see varieties like Anastasia and Savio are showing sort of 3.5, 4 on the hypocotyl root scores. So pretty much dead. Whereas varieties like Campus, they're still showing a lot of symptoms, but nowhere near as much. So there is definitely something interesting to investigate here. I also measured the root length, so this is the total root length, including all the laterals. And Anastasia at 7 days has the smallest roots of any of the varieties I tested in both non-inoculated and inoculated conditions, and also shows the biggest reduction, so 95% reduction under inoculation. Sky was a variety I was particularly interested in because it had the longest roots under inoculation. So it had the longest roots under non-inoculation as well, lost 85%, but still had quite a good amount of roots compared to some of the other varieties at 7 days. Campus, which I mentioned before as having the least symptoms, showed a 79% reduction. So that was the smallest reduction in root length. I then had a look under a microscope. So I took some of the sclerotia from the plate I showed you earlier and put them as next to, closest touching to roots grown on plates, and then took samples, stained them, put them under a microscope just to see if the pathogen at those really early stages was behaving differently next to different varieties. So if we look at Anastasia, I don't know how well you can see these images, but at 8 hours, I'm already seeing the sclerotia have germinated and they're starting to sort of produce infection structures like plaques, infection cushions. They're already starting to infect the plant after just 8 hours, which is really fast. When I looked at Sky, I saw similar things, but a bit later. So we're definitely getting the infection plaques by 48 hours, but not so much at 8 and 24. And when I looked at Campus, which is one with the least symptoms, I didn't see any infection plaques forming or sclerotia germinating up to 48 hours. So there's definitely differences in the interaction between different varieties and rhizotonia. So now looking at what might sort of underlie this in a more genetic way, I'm just going to run through how defense to necrotrophic pathogens tends to work with plant hormones. So this is the jasmonic acid pathway, and jasmonic acid is often associated with responses to necrotrophic pathogens and insect herbivory. And ERF1, which is sort of near the bottom-ish, and MIC2, they're antagonistic to each other, and they kind of form two branches of the pathway. So ERF1 and PDF1.2, that's the pathway you would expect to see for response to necrotrophic pathogens. MIC2 is more associated with herbivory. At the top of the pathway, jasmonic acid itself isn't active in the cell. It needs to work with JAR1 to become active by adding in an isoleucine. Ethylene often works synergistically with jasmonic acid in responses to necrotrophic pathogens, and it can upregulate ERF1 as well. On the other side, we have abscisic acid, which can work with the MIC2 side, and that's typically linked to abiotic stressors. Auxin does fit in with this as well, and can have effects on jasmonic acid. It has quite a similar structure to its pathway as jasmonic acid. So you see that in the presence of auxin, complexes are formed which degrade the AuxIA proteins and lead to the upregulation of auxin-inducible genes. And then finally, on the other side, antagonistic to this is the salicylic acid pathway, which is biosynthesized either through PAL4 and the PAL pathway or through the ICS pathway. We also might have activity from reactive oxygen species, which can work with the salicylic acid pathway as well. So to measure the relative gene expression, I had infected plants and non-infected plants, and I took samples at 8 hours, 24 hours, and 48 hours, and put them together in the graph. So you can see that if the value is above 1, that means that it was upregulated in the inoculated plants, and if it's below 1, it was downregulated in the inoculated plants, and it's on a log2 scale. So it's quite tricky to interpret some of these because they're done on varieties rather than lines where there's just one gene difference. But in general, we can see here that there's more activity for Anastasia, which is the bar on the furthest left. I'm not sure how big that's going to be for everyone. So Anastasia, which was our most susceptible variety, does seem to be showing more auxin activity, particularly the graph on the bottom right, which is an auxin responsive gene, is really highly upregulated to Anastasia. So this is sort of backing up the hypothesis that perhaps auxin is playing some kind of role in susceptibility. And as I said before, we know that some of the other strains of Rhizoctonia do produce PAA, which is a type of auxin and can work through the same pathway as IAA, which is the more typical one. So potentially the pathogen could be using that as a pathogenicity factor. To then look at the involvement of salicylic acid and reactive oxygen species, going down the salicylic acid side first. Here we've got PAL4, which is the top one, and that seems to show increased activity for Anastasia and Campus at 24 and 48 hours, which is interesting because you've got both the most susceptible and the most, or the least susceptible variety are both showing quite similar. For ICS1, which is another route for salicylic acid biosynthesis, it seems more clear as Anastasia. And for NPR1, it's back to showing both Anastasia and Campus. And I wondered if this was potentially because NPR1 particularly is involved in the regulation of the antagonism between salicylic acid and jasmonic acid, which I'll come on to in a second. When we look at the reactive oxygen species, we get two almost opposite graphs. So for RBOHC, which is on top, we've got a lot more activity for Anastasia. It's being really highly upregulated in the inoculated plants. And for the lower down graph, RBOHD, Anastasia is really downregulated. So that's quite interesting. Those two genes have been shown before to be upregulated in responses to different pathogens, so potentially are involved in different types of responses with reactive oxygen species. On to the last section, looking at things related to the jasmonic acid pathway. If we look at the ERF1 and PDF1 bridges, so this is sort of seeing whether the ERF1 branch of that jasmonic acid pathway is involved. At 48 hours for ERF1, it definitely seems that Campus and Sky, which are our two more resistant varieties, are more upregulated, which is what I would have expected, as that's the typical response to an ectotrophic pathogen. And when we look at PDF 1.2, you get a similar thing at 24 hours, and then Campus is the highest at 48 hours. So it's not super clear. I'm not 100% certain of this, but it looks like it might be doing that. When we look at the other side, I think it's more clear in that Anastasia, as the left-hand column on each of these, is the highest on both of these graphs for MIC2 and AAO3. So it does look like that pathway is a lot more active. To further investigate this, I grew some Arabidopsis plants, and these plants do only have one difference between each mutant. So I tested plants with ABA mutants, ethylene, jasmonic acid, auxin, and salicylic acid. And we can see here, so we've got the wild type on the far left, and 3 with asterisks are significantly bigger than the wild type under inoculation. So quite a few of my mutants were growing much more strongly than the wild type, which definitely shows that there must be some kind of involvement of these pathways, because when you stop the activity, the plants get more resistant. Another way of looking at this is to compare the non-inoculated plants to the inoculated plants, and the ones I've circled, you can see that there's very little difference at all in the size of the plants under inoculation. So that's really interesting in itself, that we've managed to stop the pathogen having so much of an impact by knocking out these genes. And just to point them out, so there's ABI4, which is an ABA resistant mutant, so it won't respond to any ABA, it's involved in the signaling. COI1 is involved in jasmonic acid signaling. AXR1 and TIR1 are both involved in auxin signaling, and SID2 is involved in salicylic acid signaling. So to conclude the work I've done so far, there definitely seems to be some differences between oilseed rape varieties, both in the symptoms they show, how root growth is affected, and also when looking at those sclerotia germinating next to the roots, there's a difference between the different varieties. Looking at the gene expression data, definitely different pathways are acting differently in the different varieties under inoculation. So there's definitely more we can investigate there. And the Arabidopsis mutants sort of back this up as well, in that by knocking out some of the activity in certain defense pathways, we can make the plants more resistant. There's quite a lot more I want to do to sort of firm this all up. I'm currently doing some investigations using plants that will show the activity of auxins, so I can inoculate those plants and see whether the auxin concentration is increasing or decreasing. I've also got some lines in Arabidopsis that will allow me to visualize the presence of jasmonic acid. And I'd like to do some ROS staining because the 2 ROS graphs were really like, I'm not quite sure what's happening there. And I hope that all of this will help us to understand the defence responses happening in oilseed rape to Rhizoctonia solani, and that will enable us to breed some resistant crops. So I want to say a big thank you to my supervisor, Rumiana Ray. Dasuni did a lot of the work preceding my PhD, and everyone in the Ray group for their support. As I said before, I'm on a doctoral training programme funded by the BBSRC and the University of Nottingham, and I'm also grateful that they have allowed me to do my 3-month placement at the AHDB, because I'm having a great time doing that too. And thank you all very much for listening.
Thank you for that. I didn't have any questions come up on the screen, but some questions from the floor. Yes, we have one over here. Got a mic coming.
Talk. I was just wondering, perhaps I missed it, but did you actually measure the concentration of the different hormones within the plant material?
No, we haven't done at the moment. So yeah, that is something I'd love to do to see whether they are changing. But yeah, we've only done gene expression data. So
okay.
Yeah, sorry.
Any other questions? Okay, well, thank you very much.
Thank you.
So our next speaker is actually not here. She's speaking from the other side of the country, so we hope this is going to work okay. So this is going to be on clubroot and looking at diversity in the UK. And Julie Smith is the speaker from ADAS.
My presentation is about clubroot diversity and the implications that that has on management. Next slide, please. Next slide, please. Next slide, please. I'm so sorry. I think we're having some technical issues. Could we go back a slide, please? Thank you. So could we go back to back a slide? Thank you. So clubroot doesn't rank nationally on the same economic level of damage as for things like FOMA, canker, and like leaf spot, but on an individual field level, it can be quite devastating. So the field on the right is would be a sort of characteristic patch of clubroot that we can often see with the discoloration of the plants. The field on the left is a field that was quite close to where I work in 2016, and you can see that it's virtually field failure. So we've published, although the damage from clubroot can vary considerably between fields within season and between seasons, we've published a relationship of 10% plants infected gives an approximate 0.3 tonnes per hectare yield loss. So it's certainly a disease to be taken seriously. Next slide, please. Thank you. So it's not just the in-season damage with clubroot that we should be worried about. There are some longer term challenges. So the literature and from our own findings, we know that climate change is likely to favour both disease development and impact on yield. So clubroot needs a soil temperature threshold of 16 degrees or above, and it needs water to be able to infect. So the slightly warmer, wetter autumns and winters that we've had have just really been, you know, almost perfect in some years for this to happen. It's got a very wide host range. We know it affects a lot of different brassica crops, including some weed species. So the carryover can be, you know, can be significant between fields too. It's exacerbated by close rotations. So we've had a few growers that over the last decade have tried to grow wheat, rape, and so forth, which has been really quite disastrous, I think, in some situations. So club root often goes undetected at the field level. We know that some of the crops are actually infected because we walk across the crops and pull plants, but the infection in that season might not be severe enough to cause above ground symptoms. So unless the agronomist or the grower is actually pulling plants, they may not realise they have a problem in the field. So it just bubbles away and increases. Molecular diagnostics, I think, need better interpretation. So we can take soil samples and we can identify club root inoculum, but what that actually means in terms of you know, the ultimate impact on yield, really the relationship isn't quite there yet, in my opinion. So the inoculum is very robust. It can survive for 15 years in the soil with a half-life of about 4.5 years. So that means that once you have infection in your field, it's very difficult to eradicate that. So clean land should really be viewed, I think, as a finite resource. We don't have any satisfactory fungicide or biocontrol options available at the moment. We've got limited management from agronomic strategies such as increasing the pH through liming or applying calcium cyanamide, correcting boron deficiencies. Those things can help, but they won't give you complete control. And we don't have really any guidance for farmers to base decisions about the short-term profitability versus protecting the longer-term sustainability of their land. So at the moment, the most popular management strategy for club root is to sow a resistant variety. But the problem with that is that our resistant varieties are based at the moment on a single major dominant gene, which is being eroded. And interestingly, there's no strategic advice to underpin the deployment of this resistance. So just to put that into context, in the UK and across a lot of Europe, we're suggesting that people only grow a resistant variety where they have to, because we're trying to protect it, we're trying to make it last as long as possible. Whereas our Canadian colleagues are taking the complete opposite approach, and they're suggesting that as soon as one field in an area has been shown to have club root, all of the surrounding fields and farms in that area immediately sow resistant varieties. So they're trying to wipe it out before it gets a hold. Some of the Scandinavian countries are taking a more intermediate approach. They're taking soil tests and they're determining the inoculum levels in the soil, and they're basing decisions about resistant variety deployment depending on what levels of inoculum they find. So there's some urgent research really needed to understand how best to deploy the resistance, because we need to make it last as long as possible. Next slide, please. So just a little bit of a look about the resistance mechanism that we currently have. I'm not a breeder, so I'm not going to try and explain in detail this slide, but the resistance that we have at the moment is termed Mendel resistance, and this originally came from crosses between stubble turnip and kale. Now, in some of the early breeding lines, I believe that there were 3 major race-specific R genes in those early lines, but only one of them made it through to Mendel. So as I said, the resistance mechanism at the moment in our current varieties is still based on this Mendel resistance, even just in different elite backgrounds, unless any of the breeders in the audience know differently. Next slide, please. So we thought there's obviously a problem. We need to make the resistance last as long as possible. But one of the difficulties is that we don't really understand our soils terribly well in the UK. So we don't know where the resistance breaking strains reside, and we don't really understand the pathotypes that can overcome this resistance, because the Mendel resistance is known to be effective against some, but not all of the club root pathotypes. So we received funding from AHDB. This was a collaborative project with SRUC, University of Sheffield, LS Plant Breeding and Soil Essentials. And the project itself was split into two completely separate parts, really. We had a very practical part, and then we had a part where we were trying to understand this resistance. So I don't have time in this presentation to talk about all of the findings from this project. So I'm just going to describe what we did under Objective 1, which was to determine the prevalence and distribution of these resistance breaking strains present in our UK soils. Next slide, please. So we took soil samples from across 90 commercial fields which we knew were infected with club root. So these fields weren't truly random in that we didn't have a sort of statistical stratification. We used our networks of farmer and agronomy contacts to obtain soil samples from these fields. And we took the soil and we carried out the traditional bioassay to test for the presence or absence of Mendel virulence. So each of these soils we grew Mentor as a resistant variety, so Mentor carries the Mendel resistance. We compared that with Tolkien, which is a susceptible oilseed rape variety, and we chose Tolkien in collaboration with LS Plant Breeding because Tolkien was at the time we thought the best match to Mentor, but given that it was a susceptible variety, and we also had a Chinese cabbage susceptible control in there as well. So it was a very straightforward assay. We maintained, a satisfactory water moisture for infection, and we kept the soil temperature at just around 21 degrees. And the plants were grown for 8 weeks and then pulled up, and the roots were assessed for the presence of club root. So we took a subset of those 90 fields. We took a subset of 30 soils in which to determine the pathotypes in those fields. We used the European club root differential set to do that. So the next slide, please. So what we found, I should just explain the graph. So the y-axis is the proportion of those sites. So this is the proportion of the 90 sites that were sampled, and the x-axis has been standardized, so we're showing the club root severity in the resistant variety as a proportion of the severity in the susceptible. So we did this because not across all of the soils we tested, we didn't get 100% of the susceptible variety infected in every single soil. So anything where infection was below, I think it was about 70%, we threw out because we felt that wasn't really a fair test, but we've standardized it in this manner. So I should just say too, the seed we obtained from the breeder rather than from commercial stock, because we felt it would be a little bit more pure. So certified seed is supposed to be 90% pure. So obtaining it from the breeder, hopefully gave us a better purity. So what we can see then is that we did have a few of the soils that, for whatever reason, even though we knew they were from infested fields, they didn't go on to develop club roots, which is the green bar at the left of the graph. And then we had a proportion of fields which developed some club root symptoms, so up to sort of 20%. Those are the hashed light blue bars. And then, but quite worryingly, we had 17% of our fields sampled developed more than 30% of clubroot in the resistant variety. And some of those fields, as you can see on the very far right-hand bar, developed between 90 and 100%, or between 90 and 100% of the plants in the resistant variety developed clubroot galls. So interestingly, there's no set threshold above which you can say that the resistance has broken down. So we set it at 30%. We spoke with other international researchers, and 30% seems to be a reasonable threshold above which you can consider the variety to have been overcome. But there isn't actually a published, internationally recognised threshold. But we thought that was a fair test. Next slide, please. So in terms of the distribution of these resistance breaking strains, we hypothesised originally that we would find them clustered in certain parts of the country where clubroots, where oilseed rape had been, quite intensively grown. But this actually wasn't the case. And we did find that these resistant breaking strains occurred really throughout the UK. I should say that we didn't take any samples from Northern Ireland, but we did sample from Scotland, England and Wales too. So that for me was quite a surprise. And it could be perhaps a reflection of the quite diverse, brassica growing history that we have, perhaps in the UK. But next slide, please. So in terms of the pathotype determination, there are three classification systems which are popular for this. So we have the Somme, which uses 3 host varieties. We have Williams, which uses 4 host varieties. But we felt the strongest classification system is the Buxari system. So this uses 15 differential hosts, which are listed here in the table. And these hosts are divided into 3 genomic subsets. So if, and you can see here, we've got the Rapa, Napus and Oleracea. So if one of these hosts develops clubroot symptoms, it's given a value which you can see in the right-hand column, 1 through to 16. So what then happens is that each score within the genomic subsets, the scores are added together. So for example, in subset 1, Brassica rapa, if ECD1 and ECD2 both showed signs of infection, you would add those scores together. So the score for that group would be 3. And the same for the other two subsets. So the pathotype designation itself is arranged in triplets, and each value is the score from each of those subset groups. So we would get a value such as 3, 1, 4, for example, if ECD1 and 2 were infected, ECD6 and ECD13. So I just wanted to explain properly how the scores were derived. And using this system means that when you look at the triplet scores, it is possible to determine exactly which of the hosts is showing signs of infestation. So could we have the next slide, please? So when we carried out these tests, and bearing in mind this is a very small subset, it's only 30 soils, I've presented here just a simple pie chart, and each of the values represents the triplet set of scores. So for example, we've got 16, 1, 14. What we see is that the pathogen is extremely diverse. We have many different pathotypes across the UK. So this is challenging for the breeders, because if you wanted to breed a variety resistant against this, which pathotype or pathotypes do you go for? Now, as I said, this is a very limited subset of samples. So I think if we tested more soils, I'm sure that we would start to pull out the dominant pathotypes in the UK. But from this preliminary analysis, it's really quite difficult to say that we have a dominant pathotype. Can I have the next slide, please? So I've just put a chart. I went into the literature and I spoke with international colleagues to put this table together. I haven't cited all of the references at the bottom because there would be too many. And it would appear, just scanning across the table then, we can pick out dominant pathotypes in certain other countries. And I suppose, unsurprisingly, the UK soils perhaps are more aligned with our European counterparts than perhaps with Canada, Australia or America. But as I said, we do have a very diverse set of pathotypes, it would appear, in our soils. Next slide, please. So in terms of breeding and looking for new and novel resistance, this is a table. We do have sources of naturally occurring resistance available to us, but when I speak with breeders about this table's been put together from various different literature. But when I speak with the breeders about this, they tell me that the resistance and these QTL are so difficult to work with, it would be almost impossible to integrate any of that into elite germplasm. So it seems unlikely that we will get new resistant varieties from naturally occurring sources of resistance. Next slide, please. So the search goes on, of course. There are some promising sources from World Gene Bank accessions. So 3 accessions here I've just highlighted in grey, as you can see, which appear to be effective against some of the common pathotypes. So the pathotypes here have been designated using one of the alternative systems, which again, that's a challenge because it makes it very difficult for researchers to properly compare findings when we have 3 different classification systems on the go. But as far as I'm aware, to date, there have been no new breakthroughs really in terms of novel resistance integrated into elite germplasm. Next slide, please. So just to summarise then, we know that Mendel resistance breaking strains of clubroots occur throughout the UK. From our preliminary and very limited pathotyping, we can see that clubroot in the UK appears to be highly diverse, which of course poses a big challenge to the breeders. We have donated the UK pathotypes that were collected in this project. We have donated them to breeding research. That was done through the Origin network. We do desperately need some research in order to guide the deployment of the the Mendel resistance, because it's breaking down and we need to make it last as long as possible, not just for Mendel resistance, of course, for any new resistance that does come onto the market. And because we know that the resistance is being overcome, and we know that the resistance isn't effective against all of the pathotypes in the UK, but we can't really rely on it as a single silver bullet. So we do need to think about longer term, more integrated management strategies if we want to continue growing profitable rapeseed in a sustainable way. Next slide, please. Thank you. So in terms of acknowledgements, of course, I would like to acknowledge AHDB for the funding. We had technical assistance from Dennis Churchill and Penny Williams, and project management and direction assistance from Tim Ball, who works in ADAS too. So thank you very much for inviting me to speak, and apologies again for the technical difficulties and for not being there in person today.
I'd like to ask her, then I suggest you email her. Okay, so we're now moving to a sort of a paper where we're looking at the practicalities of using varietal resistance in the field. Paul Gosling from AHDB is going to come and talk to us about that. Thank you, Paul.
Thank you, Graham, and good afternoon, everybody. We've heard quite a lot today about the development of resistance in oilseed rape varieties. What I want to talk to you about in the next 20 minutes or so is how farmers are choosing to deploy or indeed not deploy that resistance in the field. My name is Paul Gosling. I'm the head of the cereals and oilseeds recommended list team at the AHDB. I'm sure some of you won't know either what the recommended list system is or what the AHDB is. The AHDB, or the Agriculture and Horticultural Development Board, is a statutory levy board funded by the whole of the supply chain, from farmers through to end users such as millers and maltsters. And the aim of the AHDB is to enhance the levy payer business efficiency and competitiveness. We do that by a number of different routes. Some of them on the slide. Market intelligence, so give you information that helps growers know how to when to sell their products, their animals or their crops. We do a lot of farm level knowledge transfer activities, so that's getting groups of farmers together along with experts to discuss the problems they have and the solutions they may use to solve those problems. We do business and development skills, because a lot of farm businesses are small businesses, just 2 or 3 employees. They find it very difficult to access those sort of skills, so we help with that. And marketing is a big area for our animal sector, so we cover areas like beef, lamb and pork and dairy. And if you see an advert on the TV or in a magazine on either any of those promoting those sorts of products, there's quite likely to be AHDB money behind it. And finally, we do research and development. One area of research and development we cover is variety selection. So we have a grass and clover list of recommended varieties. We have a database of potato varieties, and we have cereals and oilseeds recommended list. And that's what I'm going to be talking about a little today. So before we can add a variety onto our recommended list of varieties, it has to be added onto the national list. Many of you probably don't know that in order to legally sell a variety in the UK, that variety has to be registered on what we call the national list of varieties. Now, a breeder will produce lots of breeding lines each year, and they will select the best of those which they think might make a new variety, and then they have to get them registered on the list. And to do that, they have to add them into national list trials. And national list trials assess 2 types of thing. They assess whether it's a true variety, whether it is distinct, it is uniform, and it is stable, so it's a real new variety, and also whether it has value for cultivation and use. So is it actually going to be better than the varieties that are available? And there are 2 years of national list testing. At the end of the first year, the breeders will withdraw the poor ones, and the rest will go into the second year. And at the end of the second year, some will fail to be national listed, either because they have a weakness, such as a very strong weakness against disease, or they're not a true and unique variety, and the rest will be added onto the national list. And it's at that point that the AHDB recommended list comes in. We take the best of those new national list varieties, we test them further in recommended list trials, and the very best of those get added to the recommended list. A farmer can grow any variety they want. It's on the national list. They don't have to grow recommended varieties, but in fact, most of the varieties they're grown are on the recommended list or have recently been on the recommended list. So the recommended list system is there for a number of reasons. It's to provide independent information on variety performance. You can find information in lots of different places, but ours is independent information. It's to help guide variety choice, but it's also to help with crop management because the information is there on resistance to different pests and disease, on agronomic characteristics. It helps growers make decisions on actually how to manage the disease and other aspects of the crop management. And the breeders actually quite like the recommended list because it validates their marketing information. They can say, look, here's our variety, here's how it performs, and here's the independent information from AHDB to show that's true. The RL can push the market to an extent in certain directions, for example, by increasing our minimum standards for pest and disease resistance that we are required to get on the list. But there is a balance to be struck between pushing the market and not stifling innovation. So, for example, if a new variety was developed which was resistant to cabbage stem flea beetle, growers would really want to grow it, so we wouldn't want to keep it off the recommended list if it had a low resistance to light leaf spot, because at the moment cabbage stem flea beetle is much more important. So what drives farmers' variety choices? Well, obviously yield is very important. The farmer needs to have enough yield, enough crop to sell in order to make a profit at the end of the day. Pest and disease resistance, yes, it's important, and it will be more important for certain pests and diseases than for others. Agronomic characteristics, so in oilseed rape, things like height, standing ability, and earliness and maturity. Farmers tend to like crops that mature early, so they can't get caught out by the weather. And markets will be important. For something like for oilseed rape, it's the specialist oils, such as the hair and the whole type varieties, that can make a difference as to whether a farmer grows them if they have a market for those types available. Cost of seed can also be important in some cases, and I'll talk a little bit about that later. And with oilseed rape, we have the variety choice between conventional open pollinated varieties and hybrids. And again, I'll mention a little bit more about that in a while. So coming back to pests and disease, these two charts show the resistance ratings for the top 5 yielding varieties on the recommended list since 2010. And the higher the number, the more resistant the variety. So if you look at the chart on the left to start with, the light leaf spot, we've got conventional and the hybrids on there. If we look back to 2010, the average of those top yielding varieties, their resistance rating was a little bit under 6. And it stayed around there till the middle of that decade. And since then, the breeders have done a pretty good job with both the hybrids and the conventionals putting better resistance into light leaf spot into those varieties. So we're now hovering a little bit below 7 on the recommended list. Situation with stem canker is slightly different. With the hybrids, we go back to 2010, the start of that decade. The hybrids weren't very resistant to stem canker. Most of the varieties were below 5. And it stayed around there towards the end of that decade. But since then, the breeders have done a very good job in improving the resistance to stem canker. And the hybrids now, the average of those top yielding ones is over 7. Conventionals is a slightly different situation. They've actually declined over that period. But generally speaking, the breeders have done a good job over that 20-year period in improving the resistance of varieties for light leaf spot and stem canker. And of course, recently they've also introduced turnip yellow's virus resistance into elite varieties. So the question is, are the farmers choosing to employ those newer, more resistant varieties to help manage their crops? What this table shows, in fact, there's 2 tables here really, there's 2 different data sets, and the reason I'm showing you 2 different data sets is because it's actually quite difficult to get good data on the varieties being grown on farm. So what we've got here is some data from the AHDB planting survey that we do every year, where we just simply ask growers what they've planted, and also some data from a leading seed merchant. What it shows is the top 3 varieties, the most popular varieties, each year for the last 4 years. And what you can see is, if you scan your eyes across the names there, what we can see is some varieties occur again and again. Campus is a variety we've heard about already today. It occurs in the top 3 varieties in both data sets for all 4 of those years. Elgar is another variety which appears every year in the planting survey data, and 2 of the years in the seed merchant data. But there are others like Phoenix, which actually only appear in 1 year in 1 data set. What I've also shown on these tables is the light leaf spot and stem canker rating, disease resistance ratings, and the ones that are highlighted in yellow are the ones that are below average for the top yielding varieties. So growers aren't choosing the most resistant varieties. They're choosing ones, the most popular ones are ones that are below average for the top yielding varieties in terms of their resistance. They're not picking the most resistant varieties. So the question is, why aren't they picking the most resistant varieties? Well, the thing that's really driving grower decisions at the moment is cabbage stem flea beetle. Anyone who works with oilseed rape knows that that's what's driving the market at the moment. We see lots of crops, unfortunately, that even if they make it through the autumn, end up looking like the picture on the left-hand side, completely devastated and very little, if any, yield coming off them. And that's had a huge impact on the options for growing oilseed rape. The graph on the right shows the area of oilseed rape grown in the UK. After many years of increasing, it peaked in 2012 at about 750,000 hectares. And since then, it's declined. And last year, we were at about 350,000 hectares, and I expect it's gone down again this year. People are stopping growing oilseed rape because of cabbage stem flea beetle. And because this impact that cabbage stem flea beetle is having on the crop, the trait that many growers are looking for now is not light leaf spot resistance, it's not canker resistance, it's actually vigor. Autumn vigor, because it allows the crop to grow away quickly and escape the damage from adult flea beetle damage. And spring vigor, because it helps the crop recover from damage from larval feeding and compensate for that feeding. But, and it's a big but, and it's why I've put real, marketed or imagined in the title. There is no standard method of measuring vigor, and no one really knows what it is. If you ask farmers, and we've done this, we've gone and asked farmers, what do you mean by vigor? You get lots of different answers. Some will say it's speed to a certain development stage, some will say it's ground cover, some will say it's height, some will say it's biomass. There is no standard understanding of what vigor actually is. And it's also dependent on a lot of non-varietal factors, things like seed size, soil moisture, and soil fertility will also affect vigor. Many people believe that hybrids are more vigorous than conventionals, and that's true to an extent, but it's certainly not always the case. And the two differential between varieties in their vigor and the actual relevance of that to cabbage stem flea beetle resistance is actually yet to be fully established. So everybody wants vigor, but nobody really knows what it is or what it does. And cabbage stem flea beetle has changed the way farmers grow the crop. Many farmers have become risk averse when they're growing the crop. And one of the aims there is to minimize the early investment in the crop. Oilseed rape, the price of growing a crop of oilseed rape is concentrated quite heavily in the early part of the crop in the establishment phase, compared with a crop like wheat, where you spend a lot of money late on when you know you've got a crop established and it's going to yield. For oilseed rape, you can plant a crop, spend a lot of money on it, and it's destroyed almost immediately by flea beetle, or it staggers through to the spring and then it fails. So farmers are trying to minimize that early investment until they know they've got a crop that they can actually, it's actually going to yield. Well, one aspect of saving money is cheap seed. Growers often want now cheap seed. And the table on the right there is some information again from a seed merchant. I've anonymized the varieties but kept the variety type on. What you can see from that chart is there's a huge range of the cost of seed. The most expensive variety on there is 3 times the price of the cheapest variety. Generally speaking, hybrids are more expensive, although interestingly, in this seed merchant, their most expensive variety is a conventional variety. But generally speaking, hybrid varieties are more expensive than conventional varieties. And for that reason, people are tending to shift a little bit towards conventional varieties. Because one way of saving money is to farm save your seed. Instead of going to the merchant and buying new seed, you use some of last year's seed. You have to pay a royalty on it, but it's cheaper than buying new seed. And of course, you can't farm save hybrid varieties. You can only farm save conventional varieties. Which brings me back to this table. If we look again at the names of the different varieties, what you'll see is next to each name is a C, it's a conventional variety, or H, it's a hybrid variety. And what you can see is the most popular varieties being grown at the moment over the last 4 years are generally conventional varieties. Campus, Aspire, Elgard, all conventional varieties, all the 3 most popular varieties grown this year. So farmers are changing the way they grow the crop to try and minimise the spend, to try and minimise the risk. So how is the recommended list system trying to help this? Well, we're trying to develop a measure for autumn vigour. This is proving very challenging. We had a protocol last year that we employed. We thought it was a good protocol. We consulted widely on it, but the trial operators, which do our recommended list trials, found it difficult to actually use, and we got very poor differentiation between varieties. We've rewritten that protocol in light of the experience from last year, and we're going to employ it again this autumn to try and get some good data. We're also trying to develop a protocol to assess the resilience to cabbage stem flea beetle damage in the spring. Again, we thought we had a very good protocol. Last year we used it. We got good differentiation between varieties, significant differences in their ability to recover from damage. We used it again this spring. Again, we got good differentiation between varieties. When we put the 2 data sets together, no significant difference. So still work to be done in these sorts of areas. And of course, there are limited budgets and increasing expectations, and the science is not always clear. Julie has already mentioned this in the previous talk. Clubroot resistance. We've got completely the opposite advice coming out of the Canadians, Canola Council of Canada, saying plant clubroot resistant varieties as soon as you see clubroot in the area, whereas in the UK our advice is don't use them unless you really have to. Completely opposite advice. So yes, we can get good indicator, we can get good resistance into varieties, we can do a lot of science on the resistance, but in some areas we're lacking the science that tells us actually how do we deploy that resistance in the field. And we heard another example this morning talking about Phoma stem canker resistance genes. We've got different resistance genes. We're very reliant on a small number of those. How do we deploy those genes in the field, and how do we persuade farmers to actually deploy them in the right way so that we don't erode the resistance which people are developing? So a few conclusions. Pest and disease resistance are important varietal traits. And the breeders have done a very good job over recent years in improving the resistance of varieties, both in their disease resistance and also introducing new traits for resistance. And these traits are more important where pesticides are not effective or are difficult to employ. We heard a little bit this morning about light leaf spot and how controlling the light leaf spot is difficult. Part of the reason for that is because actually in the middle of winter trying to spray light leaf spot when you can't get a tractor on the field because it's too wet is almost impossible. It's a polycyclic disease, it cycles over and over. Trying to control it over a long period of time is almost impossible. So that's a situation where, particularly in the north of England and Scotland, where light leaf spot is a much more severe disease, growers have become very reliant on the resistance in the varieties. Cabbage stem flea beetle has changed the way growers select varieties and how they grow the crop. But once the breeders, and some of you perhaps, get some resistant or tolerant varieties to cabbage stem flea beetle, then other traits will once again become important. Things like clubroot, things like verticillium, things like light leaf spot and Phoma. Thank you.
Thank you, Paul, for that interesting talk. So, are there questions? There's some interesting thoughts there, and yeah, the way people growing oilseed rape view it at the moment, and this sort of dramatic drop in the amount that's being grown in the UK because of cabbage stem flea beetle. Anyone got some questions? No. Okay, thank you very much. So, cabbage stem flea beetle is where we're staying at the moment. So our next talk is on companion plants to reduce cabbage stem flea beetle attacks in oilseed rape. We have Gaitan Samandi Korda to talk to us about that. Thank you.
Good afternoon. So yeah, I will present some research that we are conducting at Rosenstead Research with Sam Cook as part of the project called EcoStack. And the focus of this presentation will be on companion plants to reduce cabbage and flea beetle attacks on oilseed rape. So we all know that the oilseed rape is an important crop in Europe and in UK, but since the ban of neonicotinoids its treatment, we have increasingly more problem related to insect on oilseed rape. And this is because oilseed rape is attacked by many insects. We have insects that are attacking the plant in autumn, such as the flea beetles, the stem weevils, but also insects that are attacking the plant in the spring with the pollen beetles, the seed weevils, and the midges. So the focus of this talk will be on the flea beetle. So it's an insect which is now considered as the main insect pest of oilseed rape in UK. And as we saw in the previous presentation, we have a reduction in the oilseed rape harvested area in UK since 2012, which is partly related to the strong infestation we have in flea beetles, and especially in the south of the country. So the problem with this pest is that we have an adult which is attacking the leaves of the plant and which is causing damage to the plant, especially when they are small. And the larvae which is coming later in the season and develop into the stem and in the petiole of the leaves later in the season. And we are at the stage where without the neonicotinoids treatments now, the pesticide alternative that we have, such as the pyrethroids, have a lot of resistance. I mean, the insect develop a lot of resistance against those compounds, so they are less and less efficient, and there is not yet other option available now. And if we look to other strategies, such as biocontrol, for example, we have not a lot of knowledge on larval parasitism, and we have some cue that there is some adult parasitism, but the level of parasitism we observe is relatively low. So basically, we are now in a situation where farmers don't have a lot of options to control this insect. And one of the options that could be useful for them is to use companion cropping. So basically, the companion cropping is when you are growing at the same time simultaneously different crops in the field. And these have been shown to be beneficial for ecosystem services in the crops, such as with suppression or disease control. But also, there is a link between the plant biodiversity in the field and the herbivory we observe. And this is because the companion plant can disturb the host plant location by the insect, by hiding the crop, or by attracting the insect outside of the crop, or by repelling the insect. And the companion crop can also be used as have a positive effect on natural enemies by providing them some resource, for example, or secondary host. So now we have more and more reports from farmers in UK who are trying to develop some companion cropping systems by trying different things, such as clover, buckwheat, and veg or cereal volunteers in the oilseed rape. So this comes from observation made in other countries in Europe, where they use frost-sensitive legumes. And these have been shown to be able to bring some nutriment to the crop, but also to reduce flea beetle attack and stem weevil attack. But the problem is that this kind of strategy is not practical in UK because the winter are not cold enough, and so they don't always destroy the cover crop. So we need now to identify companion plants for oilseed rape that are adapted to the conditions in UK and be able to show to bring robust evidence that they are effective against the flea beetle. So what we did at Rosenstead is that we conduct different field trials over the last 3 years, where we test a variety of combinations of oilseed rape with different crops, such as white mustard, clover, Bersim clover, beige, turnip rape, which is used as a trap crop, because it's attractive to the flea beetles. We also try some straw mulch, which was used to simulate direct drilling into the stubble, and also different cereal, because we had report that cereal volunteers can have a positive effect in the control of this insect. So we conduct experiments at Rosenstedt, but also in Germany with University of Kassel, who replicate some of our experiments. And on this experiment, basically, we measure the adult feeding damage on the leaves, but also the plant density and the larval infestation. But I will focus my presentation mainly on the adult feeding. So here you have some results of the first field trial we conduct. So on the side, you have the proportion of leaf damage by the insect according to the different dates where we did the samplings in September and October. And in red on the top, you have the oilseed rape control, where it's only oilseed rape. And there was two treatments, so the oilseed rape mixed with white mustard and the oilseed rape mixed with wheat, where we observe a reduction of the flea beetle attack, clearly. We didn't saw an effect of the clover, but at this stage, but it was probably related to the fact that the implantation of the clover was relatively bad this year. So after we did another experiment where, because we saw that the wheat had a positive effect, we wanted to test different cereals in combination with the oilseed rape. So we test some barley, some oats, some rye, and some wheat, and also a straw mulch to simulate the direct drilling into the stubble. So what we observed during this experiment is that we had a reduction only with the addition of straw mulch. So this was probably related to the fact that this season was very dry, and the mulch was like keeping some moisture in the soil and was allowing the plant to compensate better for the attack of the insect. After, we didn't saw significant differences between the other treatments. But we choose for the next experiment to continue working with a cereal on oat, because the oat seemed to be a better option because it was the seeds were less expensive and they were easier also to kill with the herbicide. So then we replicate this experiment in Rosenstedt, but unfortunately the experiment failed. But we replicate it also in Germany. In Germany, they made an experiment with 3 different treatments. They tried the oilseed rape, the oilseed rape with oats, and the oilseed rape with mulch. And they saw a clear reduction of the flea beetle attack, both with oat and even bigger with a straw mulch. So which was like confirming that those companion strategy were potentially working. And for the last experiment that we conducted, we tested some clover with vetch, oilseed rape and oat, turnip rape, as I said, as a trap crop around the plots of oilseed rape, and different control. And what we observed is that we had a very good effect on the reduction of the flea beetle attack with the combination of oilseed rape and oat. A good effect also observed with a berseem clover, but no effect on the turnip rape. And what we saw also is that we directly had a significant effect of the oat very early in the season, while the effect of the berseem clover was observed later in the season. And this was probably due to the fact that the clover is taking more time to develop, and so potentially to hide the plant or something like that. But after, we wanted to go a bit more deeper into those observations. So on this experiment, we wanted to see if like the reduction of the damage we observed was related to the density of the cover of the different companion plants. So what we did is that on the plots of the experiment where we had oilseed rape and clover and oilseed rape and oat, we took some small plots and we took all the plant biomass and we weighted this biomass, and we tried to find if there was a relationship between the damage level we observe and the biomass of the companion plants. So the illustration we have on the left, you have the mean level of damage on the oilseed rape compared to the dry weight of the companion plants. For in red, the treatment with berseem clover, and in blue, the one with oat. And what we observe is that the more we have clover on the plots, the less we have damage of flea beetles. And this relationship is not true for the oat. So this is also probably because the oat was sown at a higher seed rate than the clover, and even at a lower density of oat, the density was still pretty high, and so was probably already having an effect. But interestingly, if you look at the illustration on the right, you can see that the more we have berseem clover into the plot, the bigger the oilseed rape plants are, and this relationship is the opposite for the oat. So which means that we have a competition between the oilseed rape and the oat. So in conclusion for that, we saw that the more we have clover, the more the plants are protected and the bigger they are. And the oat is potentially protecting the plants, but also compete with them. We also did some larval infestation measurement on the different experiments, but I'm not going to come in the detail of that to not take a lot of time on that. Basically, what we saw is that there is a trend toward reduction of the larval infestation when we have companion plants, but this is not always true, because the problem we observe is that also the companion plant can make the plant bigger, and when the plants are bigger, they're also more attractive for the insect to oviposit on them, and the larvae can potentially also develop better in those plants. So it's like a mixed effect that we observe, and we are still trying to work on that to try to understand it. So in conclusion, we saw that the addition of companion plant to oilseed rape can reduce flea beetle attack. We saw that serial volunteers or sowing oat, for example, as we did in our experiment, can be a good option to reduce the attack, but it needs to be destroyed early in the season to not compete with the oilseed rape. We also saw that the clover can reduce the feeding attack, but the problem with the clover is that it takes more time to grow, and so doesn't protect the plant very early in the season when it's very susceptible to the flea beetles. And that for the larval infestation, there is still like some work to do, because we're not sure how it's going to affect the insect. So the question after is where to go from that. There is probably clearly a need to go toward a screening of a larger diversity of companion plant and test different species to identify which ones are the most efficient to control this insect. If it's an agrochemical practice that need to be used by farmers, they also need to try to find what is like the best seed rate, both for the oilseed rape and the companion plant, how to control the companion plant, this kind of thing. And the work we did was mainly experimental, so there is still a lot of work to do on that. There is a need also to understand what are the mechanisms behind that, the effects we observe. Because at this stage we don't know if it's just the plant which is hidden by the companion plant, or if there is like any kind of repellents or something like that that can be played by some species and not others. And finally, we don't know what is the effect on other pests and on natural enemies, and is it like favoring them or on the opposite, being detrimental for them. So that's still questions that we need to work on for later. So I would like to thank Sam Cook and Todd Jenkins, who are working on EcoStack, and also all the people from Mozambique Research who help us for this project, as well as Sasha Krishner and Julian Winkler from the University of Castle, who help us for this experiment. And this work is part of a project called EcoStack, which is funded by H2020 and the European Union. Thank you for your attention.
So question just come in as I was being run. One or two of them have been coming in a bit late, so in between me and up here. But this one was asking about the effect on yield using your companion crop.
Well, we are not sure yet. The problem is that we have yield data only for the first year where we did the experiments. The year we had after, even if we saw a reduction of the attack, the problem we had was that the implantation of the crop was not very good. And so at the end, we had to destroy the experiment at the end. But this was partly related to the fact that we had a lot of attack from the field beetle, but also because the drilling was not made in the good conditions, and so the implantation was not very good. So we don't know yet really how it affects the yield. Yeah,
still to find out. Yeah. Any questions from the floor? Yeah, there's one up over there. It's a bit of a walk for you. Yeah, but they won't hear on the video if you do that. Yeah.
Thanks. Just to ask if you have any knowledge about what farmers are doing in the field at the moment in terms of deploying these techniques.
Well, we know that they do it. If you are following, for example, farmers on Twitter, they quite often post some pictures where they say that they try these kind of things. The problem we have now is that a lot of farmers are trying a lot of different things with a lot of different species and a lot of different seed rate, for example. And we did a bit of sampling with farmers, but the problem is that it's very diverse. So we are at the stage where it's difficult to get like consistent result from that. So the thing we wanted to do on our side was really to show that if yes or no, it's something which is having an effect, to after leave the possibility for the farmer to try to find, okay, what is going to be the best option for them in their own situation. Because the problem we have now also is that a lot of people try and they see that it doesn't work or that it works and they say, well, it's great. And so somebody try and say, oh no, it's really bad. I shouldn't have tried that. And because we are at the stage where we don't have like proof that it's working or not.
Okay, any other questions? Okay, thank you very much. So final speaker for this afternoon, Bruce Fitt. I'm sure this morning, if you're here this morning, you will have seen his name pop up on acknowledgements on a lot of the presentations and also on some of the posters as well. And Bruce is going to talk to us about climate change and oilseed diseases and various aspects of that. Thank you, Bruce.
Well, thank you very much, Graham, and thank you to AFCP for inviting me to give this talk. So climate change is a fact. We've had an increase in carbon dioxide concentrations over the last 60 years. And coming nearer to home, this is data from Rothamsted, just a few miles away from us. And whilst the mean temperature was stable over a period of more than 100 years, over the last 30 years, we've seen a gradual increase in mean temperature. And climate change threatens food security, particularly for those areas of the world coloured in red or brown, where they may suffer more than a 25% loss in agricultural productivity. And that means that people don't have enough to eat. I was at a conference where Lord Stern suggested that if we didn't do anything to mitigate effects of climate change here, then perhaps all those people that live in countries where they are suffering reductions in agricultural productivity should move here. And crop diseases also threaten food security. These data for 4 major world crops suggest that even with crop protection strategies in place, crop diseases are accounting for £150 billion worth of losses each year. And without any crop protection, then they may account for £270 billion worth of losses each year. So the challenge for us is that if crop losses could be decreased by 1%, an extra 25 million people would have enough to eat without any extra use of resources. So turning to oilseed rape, we've seen this graph several times already today, and we've heard a lot about light leaf spot, the main disease problem on oilseed rape, and fomer stem canker. Farmers in the UK can afford to use fungicides. So why don't we just control the diseases with fungicides? The problem is the variable pathogen populations. For example, the light leaf spot pathogen, Paranopozizib brassicae, is developing insensitivity to many of the fungicides that are used by farmers. And so the farmer might spray the crop and get no control whatsoever of disease. What about crop resistance? We've heard quite a lot about breeding for resistance, and ever since man started to grow crops, there's been selection of plant material that's more resistant to the diseases that attack those crops. But again, there is a problem with the variability of the pathogens that cause those diseases. On the left, we've got a resistant crop grown in Australia. This is resistant to the fomer stem canker pathogen, Leptosphaeria maculans. On the right, we've got a crop where a resistance gene has been bred into that crop from a wild relative of oilseed rape over a 15-year period. And then when the crop is grown on a wide scale in Australia, the pathogen population changes to overcome that resistance, which is effective initially, and farmers are suffering 90% yield loss. So today, I want us to think about what impact does climate change have on crop diseases, to think about adaptation, how can we be prepared for future impacts of climate change on disease epidemics and food security, and mitigation. Can crop disease control help to reduce greenhouse gas emissions now? Now, you get a severe disease where you have a susceptible crop, a virulent pathogen, and a favorable environment. It's obvious that changing the climate affects the environment, but climate change can also affect crop resistance and pathogens. So turning to work on the impacts of climate change on oilseed rape. What we did, we combined a crop growth model, which gave us yields with simulated weather for different climate change scenarios. Comparing the baseline 1980s in the left-hand map with 2050s under high carbon dioxide emissions on the right-hand side. And we can see that that crop growth model, which assumes that diseases are controlled, combined with predicted climate change, we'll get an increase in oilseed rape yields, particularly in that area in the red, in Aberdeenshire in Scotland, which is one of the oilseed rape growing areas in Scotland. But that is ignoring the impacts of climate change on disease, and in particular on this disease, which we've already heard quite a lot about today. Now, we developed a model to describe the progress of the epidemics. We've already heard about the life cycle, but this considered the life cycle in 3 stages. Firstly, on the top right, we've got the leaf spot stage, and that is dependent, the timing of leaf spotting is dependent on the temperature and rainfall over the summer period, when the ascospores are maturing and when the crops are getting infected. And then the second 2 phases are dependent on the temperature and the resistance of the cultivar. We developed this model to help farmers decide when to spray their crops in the autumn. We've heard earlier, you have to spray the crops in the autumn. It's no good waiting till the pathogens got into the stem, it's too late. And so this is on the Rothamsted website, and growers can look at a map and look at the date when some 10% of plants in their crop may be affected by the disease, which is the spray threshold that's used. Now, we developed that weather-based model of the epidemics to help farmers, but it can also be used in climate change predictions. So we've already heard about incorporating a crop growth model with climate change predictions. We now incorporate a disease model and include a yield loss model. And when we do that, we find that for susceptible cultivars in southern England, there would be a 50% loss in yield. So it wouldn't be worthwhile growers growing the crop unless the disease is controlled. I talked about resistance. Now, resistance against Leptospira maculans, we've already heard something about it before today. There's the R gene mediated resistance, which operates at the leaf spot stage of the disease. And then there's the quantitative resistance that operates as the pathogen is growing down the leaf stalk and in the stem tissues. And both of these can be affected by climate change. This is some work done by Yongju using material provided by French collaborators at INRA Rennes, and that's material near isogenic material with or without a particular resistance gene, RLM6. And you can see that at 15 degrees, the resistance is effective. The Darmor without the resistance gene develops leaf spotting. The pathogen infects it. But with the resistance gene, there's no leaf spotting. However, if you put the temperature up to 25 degrees, we find that the resistance gene is no longer effective. So that resistance gene is temperature sensitive. And Yongju's already mentioned using L-maculans labeled with green fluorescent protein, and you can show that where there's no leaf spots, the pathogen does not reach the stem tissue, and so there's no canker, no severe disease. Effects of temperature on quantitative resistance. In this instance, the leaf stalks were inoculated and then cankers on the stem were measured. And you can see that at 15 degrees, the amount of disease that develops on Urole without quantitative resistance is much greater than on Darmel. But if you increase the temperature, the difference disappears. However, one thing we need to bear in mind when considering impacts of climate change on disease epidemics is the uncertainty in predictions. And in particular, all the early work on impacts of climate change on diseases was done where pathogens were grown on artificial media. And this work is part of Faye Newbury's PhD thesis and shows that if the canker pathogen is grown on artificial media, it grows best at a temperature in the high 20s, just below 30 degrees. However, she also did the work with plants inoculated with the pathogen at a range of temperatures. And here we found that the optimum temperature for producing disease is much lower. So this shows us that we have to be careful when we are making predictions to use appropriate methods of measuring the disease. So to conclude on impacts of climate change, we predict there'll be an increase in oilseed rape yield with climate change if disease is controlled. We predict that oilseed rape yields will be halved in susceptible cultivars if disease is not controlled. And there is uncertainty. One cannot base predictions on experiments done with artificial media. They've got to be done in plants. So our predictions can be used to guide strategies for adaptation. For example, we need disease resistance that operates at higher temperatures. Breeders need to trial their cultivars that they hope to introduce into the UK in a few years' time in countries where the temperatures are currently higher. We do need information on other crop diseases because such predictions can guide government and industry forward planning by identifying diseases that will become more important to ensure future food security. And this morning, the Adaptation Committee of the Climate Change Committee, which advises the UK government, published a report, and that suggested that in the UK we're very ill prepared in terms of adaptation to climate change. All the debate is about trying to reduce the increase in temperature, but we know that climate change is going to happen, even if the amount that the temperature increases by is reduced. And we need to be prepared for that, and we're not. And thirdly, I want to go on to mitigation. Can crop disease control contribute to climate change mitigation? The government has got a target to reduce greenhouse gas emissions from agriculture and to try and have net zero greenhouse gas emissions by 2050. The NFU's got a target to reduce greenhouse gas emissions by 2040. The problem is sometimes that there is a conflict between providing food at a price when the most vulnerable in society can afford to buy it and reducing greenhouse gas emissions. If we look at oilseed rape, to produce 1 hectare of oilseed rape, most of the greenhouse gas emissions are associated with the use of the nitrogen fertilizer, both in the manufacture of that nitrogen fertilizer and when it's applied to the crop. The nitrogen fertilizer is broken down by bacteria to produce nitrous oxide, which is 300 times more potent as a greenhouse gas than carbon dioxide. So using some data from experiments done at Rothamsted and by ADAS, we estimated that if you control diseases, in this case light leaf spot and canker, on oilseed rape crops and increase the yield, then per ton of oilseed rape produced, you save 100 kilogram equivalents of CO2. And other work, similar work has been done by colleagues at ADAS on wheat, and we did work on barley, and the estimate is that each year, by controlling diseases, one could save more than 1 million tons CO2 equivalent by controlling crop diseases in arable crops in the UK. So with disease control, with crop resistance, and it's much more difficult to measure that, or with fungicides, one can decrease greenhouse gas emissions. And these greenhouse gas emissions can contribute to UK targets for reducing greenhouse gas emissions. But ideally, we need arable crops that don't need nitrogen fertilizers. Legumes like peas and beans have got nitrogen fixing bacteria associated with their roots. So if we could produce other crops that didn't need nitrogen fertilizer, that would be an excellent way of reducing greenhouse gas emissions. So we need information on impacts of climate change on crop disease losses to guide strategies for adaptation. We need accurate assessments to show how improved crop disease control can contribute to climate change mitigation. And we need vigilance to maintain or improve crop disease control despite changing pathogen populations and a changing climate to ensure global food security. I would like to thank many people who've contributed to this work, and it's good that I know that many people are watching this presentation virtually, and also I know that some of the funders from DEFRA and HDB are also watching this virtually. So thank you to all of you. And at this AFCP meeting, I would particularly like to thank the agricultural charities who've contributed to our BBSRC Link project and have in part funded many of the PhD students who've worked with me over the years. I would like to thank colleagues at Rothamsted who worked with me, particularly in the plant pathogen climate interactions group. And again, it's good that many of you are with us virtually today. And also to thank colleagues at the university, and particularly members of the Crop Protection and Climate Change group. And I'd like to say a special thanks to Avis Hall, who masterminded my move to the university, and many other colleagues that I've worked with at the university over the years. And this autumn, I'm proposing to relinquish some of my administrative duties at the university, though I hope that if the powers that be at the university agree that I can continue with some of my research, particularly the research on climate change impacts, adaptation, and mitigation. Thank you very much.
Thank you, Bruce. Other questions? I was wondering, looking at obviously something like producing new varieties for changing climate. Obviously, if we're looking 20 years ahead, then those have got to start being produced now. Is it happening?
Yes, exactly. You've got to think ahead.
Yeah. So is it happening?
Breeders do plan quite a long way in advance, but judging from the report that was issued this morning, perhaps government doesn't.
Okay. There's a question up there by the look of it. Yeah. Obviously, the person up there wants to keep you fit, running up and down the stairs.
Thanks, Bruce. That was a masterful talk, which I really enjoyed. One of the, just to go back to the broader picture, one of the suggestions that's circulating a lot in policy debates around food security and our future towards net zero is that we need a greater diversity of cropping, of types of crops grown in this country. Well, we need them internationally because we're heavily dependent on just a small handful of key food crops. I just wondered if what your thoughts on that are, and presumably each new crop anyway brings a new pathogen challenge.
It certainly does, but I think that it is likely that there'll be a more diverse range of crops grown. Yes, they will have their own pathogens, but if you grow large monocultures of exactly the same crop all the time, then that's ideal from the point of view of a pathogen.
For a start, perhaps.
Well, one of the problems with peas and beans, and maybe we've got people here from HDB, so if anyone else wishes to, it's the yield isn't always very stable, I think. Beans have a problem with chocolate spot, not if we've got weather like we have today, but maybe if we get the rain that we've got tomorrow, that favours chocolate spot on beans. I've certainly worked on beans before, and they can get quite hammered by disease. I'm not so familiar with peas. If we had our virtual audience here in person, I know someone who's done a lot of work on peas, but I don't know whether, Paul, whether you want to add anything.
Yes, I think Bruce is right on the peas and beans. Beans in particular are notoriously variable in their yields. At the moment, farmers tend to grow the crops that make them the most money. And in the UK, that's wheat. And that's why we have so much wheat in the rotation. And that's why farmers will go back to oilseed rape if we can sort out the cabbage stem flea beetle issue, because it is the most valuable break crop for them. If we want farmers to grow a more diverse range of crops, I think there will have to be government incentives to encourage them to do that. Otherwise, they will tend to just go for the ones that make the most money.
Yeah. Any other questions? Okay. Now, Bruce is line manager. Professor Mike Page wants to say a few words.
Thank you very much. My name is Mike Page, and I'm the Associate Dean Research for Life and Medical Sciences, which is the school in which Bruce does his work. And because I was aware that Bruce was going to, how did you put it, stepping back from administrative duties, it was a nice description. I'm not sure that I represent a power that be. Having spent a year in lockdown, I've realized I don't even represent a power that be in my own house, let alone anywhere else. But I think I'm the closest that you'll get today. So I wanted to say a few words of thanks to Bruce for everything he's done. Just a little bit of history, which I managed to extract from him. He joined the university on the 4th of January 2011, the same day as the Vice Chancellor of the university, Professor Quintin McKellar. And he's been, as you know, Professor of Plant Pathology ever since. Almost immediately, one of my predecessors then inveigled him to set up his research group and then to become research lead for the department. But such are the number of administrative changes that we've had ever since, neither of us could quite remember what the department was called. They seem to change their name every couple of years, and we couldn't recall it. But it wasn't a completely outrageous name, and you can presumably supply it yourself. David Barling, Professor David Barling, joined then to run the Centre for Agriculture, Food and Environmental Management. And that included Rothamsted, as you know, RVC, Royal Veterinary College, and Oaklands College, I think, at the time, CAFM. And more recently, CAFM has turned into CAFM Research Centre, which sits within Life and Medical Sciences, which is why I'm here. And David and Bruce are co-directors of that. It's one of the administrative duties that Bruce has done so well. Bruce did disclose to me that as the head of agriculture, food and veterinary sciences, he was required, or maybe it was a great pleasure, to meet many of the great and good of Hertfordshire rural life. And he did let on that he had to pretend to be more interested in horse racing than perhaps he actually is. His preferred steed is his 1966 Morris Traveller, of which he's inordinately proud. And I thought that I could maybe construct a speech comparing Bruce, you know, the classy performer, eschewing flashy technology in favour of a sort of timeless elegance. But then I found out how often the university security had been called upon to help remove it from the campus. I thought it was probably a metaphor that I shouldn't pursue too far. So I failed on horses and cars. So I've settled on ships, 3 ships, but not the sort of ships you'd think. Bruce, we're very grateful for your scholarship. It's very evident today and in all the interactions you've had with people here and the many people watching. For your leadership in two REFs, Research Excellence Frameworks, that you've taken us through so very ably, as I know, particularly since one has just been submitted, and your leadership with David of CAFM Research, and your friendship as well. When I had a loss in my family this year, Bruce was one of the most solicitous and giving of all my colleagues, and I thank you for that. You're a very fine colleague and a very fine human being. So we're not losing him entirely, I'm very pleased to say. With your work on climate change, which you've just outlined for us, I think you can add stewardship to your fleet of ships. And we very much look forward to working with you in the years to come. We can't really thank you enough, but just to make a start in thanking you, I've signed all those timesheets that you brought in for me earlier. So having met you now for the first time in 3D for almost a year. So anyway, Bruce, we thank you very much for all that you've done, and we wish you every success in the future. Thank you.
So if I could just add my best wishes to Bruce. I've known him for more years than I care to remember, really. Go back a very long way from when I worked at the Plant Breeding Institute, as it was in those days. It was a long time ago. So to you, those of you here, those of you that are watching online, we hope you've enjoyed today and that you found it very interesting and informative. Do get back to people if you have other questions. I apologize, one or two of you asked questions which came in a bit late. As you won't be able to see online, my computer's in a slightly different place from where I am when I'm standing up here. So if you didn't send the question till I was walking up the stairs, you know, it came in a bit late. But sorry about that. And maybe you could contact the speaker if you want to know the answer. So firstly, obviously, I'd like to thank all the speakers today for their excellent work. I apologize, especially to the first speaker, for the problems we had there with the sound, etc., which I think confirmed me in my belief that actually having people here would probably work better than trying to do it all online. It was also really great. It's the first time I've been in a meeting like this, even if it is not that many people actually present for a long time. And I'm sure that's true for you as well. So it was really great to actually be here. And thank you to those that turned up here in the auditorium. Secondly, I'd obviously like to thank the university. They've worked very hard for us to be able to do this, to provide this COVID-free environment, this safe environment for us to be in, to jump through all the hoops which were needed, etc. It's been a lot of work for them, so I very much appreciate it, and for the projectionists and so on for getting the slides up and running. So don't forget to look at the posters. You probably haven't done so yet. Certainly those in the auditorium. I'm sure you weren't, because you're out sunning yourselves at lunchtime. But do have a look at them. They're all on the AFCP website. You've had the link, and if you can't remember what it is, then contact me or somebody else and have a look at them. There's some very interesting stuff there. The presentations have been recorded. I think most people have agreed to that. I'm not sure quite exactly if everybody has or not, but so they will be available if you want to see them again, or if you know other people that might be interested. And as the speakers know, we're also planning to publish much of the work that's been done today, where people are asking them to write something up, whether that be long or short, and we'll put that together hopefully, well, during the summer anyway, and produce some sort of proceedings from that. So we'll let you know when that has been done as well. So just up to me now to, I think, to wish you, those of you here, a good trip home, and hope that we might all be able to meet together again before too very long, and perhaps be able to actually sit a bit closer together than we can today. So thank you very much.